All right, we are live. Good afternoon, everybody. Also, good evening. It's 4:50 p.m. right now on RV Falcor 2. Welcome aboard. My name is Sophia. I'm the multimedia technician, and I have the honor of bringing you the stream today. Right now, we are at Desecheo Ridge, looking at some more deep-sea corals. We are live from RV Falcor 2, and this is live from ROV SuBastian. This is our 504th dive. And if you have any burning questions for our deep sea coral experts, please drop them in the chat over to your right. And without further ado, I'm actually going to introduce them. So take it away, gentlemen.
Hi guys, everyone who's listening. My name's Sam Bozen. I'm a postdoc researcher at Lehigh University, and I study coral microbes, or microbiomes. I study symbionts, bacterial and otherwise, in corals, figure out their impact on coral health.
Hi everybody, my name's Scott Wonkel. I'm an isotope biogeochemist at Woods Hole Oceanographic Institution. There's lots of us here from both Lehigh University and Woods Hole. Most of us from Woods Hole here are from the marine chemistry department, and so we're really interested in this intersection between deep sea biology, biological communities, and the chemistry that both supports them living here as well as how they impact their surroundings themselves. So we've got lots of tools on board today. We have a novel sensor called Solaris here that was developed by Colleen Hansel's group, also at Hui, to measure reactive oxygen species, and others of us are also interested in trace metals and nitrogen and carbon cycling in the context of all these fascinating deep sea environments that we have here. So we're going to be taking a long look today at some deep sea corals and sponge communities and sampling, and yeah, as a geochemist, I promise not to babble on too much about the biology, as that's not my strong suit. I'll defer to Sam or Nicole or anybody else here for that. Colleen Hansel's at the helm over there as the science lead. Say hi, Colleen. We got Alex and J-Rod at the controls of the ROV today.
Right now we're circling this Calagorgia colony so we can later create a 3D model.
So Sam, will they, just by the ROV circling around like this and collecting enough high-res imagery, that will be able to be used to piece back a 3D image?
Yeah, we hope so.
Okay.
Nicole knows better about that. We're still testing it out.
Is it like a special software that does this?
Yeah, I forgot what it's called. There's one that Nicole was using called Meshroom.
Okay.
But there was one or two other ones they were using that had similar performance, or maybe even better.
The opioids, yeah. So, are you guys going to make a Solaris measurement first?
Something like about like this?
Yeah, I know. Okay.
Okay. All right. Sorry to go around the other way that I just did. Nice and slow, right? Yeah, nice and slow. Try not to pull. Maybe out another 5.
What do you think that is? That a black coral? No, this bright neon green one. Yeah. What do you think?
And when we get a chance, it'd be nice to zoom in really tight on a lot of that micro, the small scale stuff on the rocks here. I think there's a lot going on there.
Yeah, there's a lot of coral species here.
I know the Caligorgia is the charismatic megafauna.
Yeah,
of the,
that looks
of
like
the,
one of the biggest colonies, biggest species right here at this location.
But there's crinoids there we can see, and black corals. Is that the bottle brush?
It's a bottle brush
Yeah.
growth form, black corals.
Stylopathes.
There's
You're the
probably a
black
few
coral.
different
Jeremy's
species.
the resident black coral expert here. Which ones? The bottle brush. Like many worms?
Like one or two.
Really?
Like modify the corallum by bending the branches inward to make the worm run to protect themselves from predators.
Really?
And we never find those black coral species without a worm. So there's a lot of questions about.
Is it a symbiosis?
Yeah, but it doesn't benefit the black coral.
Okay.
But it benefits the worm.
Okay.
So it's a one-way.
A parasitism? No, I forget. Is it,
I mean, I don't know if it's is it
or is it right? Is it like a bird nesting in a tree?
Well, it's harmful because the branches are how the coral feeds. So if you pull its branches inward,
It's not feeding.
it's feeding less.
Yeah. Interesting.
Maybe there's some benefit that we just don't know
Maybe.
in certain contexts.
Oops. It's off screen now. There's a little, is it a hydrocoral?
Oh,
Tiny, it's like right up here. No, not that. Again, I'm Scott and I'm a geochemist, so I'm practicing my coral identification here with these experts. Like this little guy, this little white guy here on the left.
oh, it could be.
Is that a hydrocoral?
It could be a little hydrocoral. Or sometimes bryozoans can look like that.
I'd be curious to know what all of that pink
Yeah.
stuff is
I was
on the
wondering that
bottom
too.
and all the yellow things. Like,
pink
are
s-
yeah.
Yeah,
The
Colleen
pink
was,
stuff is probably crustose coral and algae, which is evidence that there's enough light at this depth for photosynthesis. It was pretty deep, right? 175 meters.
175 meters. That's nuts.
Yeah, those look like they might be little tiny lace corals. The yellow, I'm not sure, but it could be sponge. Oh, yeah, there's a bunch of brittle stars,
Ophiuroids?
ophiuroids, yeah, all over Caligorgia. We see that in a lot of corals, a lot of coral species, especially the. Yeah, that's good.
Hydrocoral identified, asterisk, by the geochemist. Questionable. Yeah, so you think that's coralline crustose, and that's crustose
Yeah, I
coralline
think so. I
algae?
think the pink is, yeah.
It's a lot of it.
hmm
So I guess they must not need much light. I mean, 170 meters is pretty.
Yeah, I think the deepest is like a little over 200 meters, and I think it's also a red alga.
We're seeing Solaris at 150 meters outside the threshold.
With
Beyond that,
photosynthetic symbionts.
right,
Yeah.
beyond, and they were as flat as could be. They were like.
Oh, the Agaricea? Is that what they are? The big plates?
Yeah, there's aquaporum.
They're acropora. You say acropora? I grew up saying that too. And everybody at Hui says acropora. And I get made fun of for that.
They don't
See.
apparently they'll also say.
Ah, okay, good. I don't feel so bad now.
But yeah, it was like at 150, we're seeing them really, really flat, maximizing.
Yeah, of course, yeah. In some of the video surveys from the south side of Puerto Rico, when we were researching this area, there's, I think, what are Agaricea, and they're these big flat plates. Again, that growth form that maximizes light. And as you move up that same species, as you move up in the water column, the same species growth form looks different. Yeah, the morphology, I guess. Yeah. So Sam, do these guys get infected? I mean, there's some dead branches there. Do they get infected by the same kinds of diseases that affect shallow water corals?
Not much is known about the diseases that affect mesophotic and deeper species of coral.
Okay.
A lot of those diseases in shallow water, like the new ones, like Skittles,
Yeah.
aren't really, I don't think they've been observed in deeper waters.
Okay.
Some of that damage could be caused by physical
Grazing.
injury.
Oh, okay.
Yeah, or grazing. Like these corals are pretty brittle, this species. It'll snap readily.
Okay. I think so, yeah. I think that's... Colleen, are you setting up for Solaris after this? On this one? Okay. Yep. Grunt? Is it a grunt? Yellowtail grunt? No? Yeah. I used to love trying to identify tropical fish, but I'm out of practice. So this guy over here, is that a...
I'm not sure.
This is the Calagorgia.
Yeah, the one here that looks like kind of like feathers. The one behind it's different species of coral. I'm not exactly sure what it is. Hopefully we get a closer look at it.
I mean, as far as the Solaris measurements go, we could sit here and make measurements of 20 different species, I think. Yeah, exactly. This one, the big, is a Calagorgia. Where'd that cheat sheet, the video guidebook? Oh, you've got them? Are there multiple copies? Or is there only one set? Okay. I just want to take a look at some of the pictures so I can, again, become an expert by the end here. Exactly.
Yeah, I was saying to Sam, like, I was looking up, like, the morphology of different groups to try to add it to my guide, and it's like, no, this isn't very known or studied, like, more research needs to be done on, like, every single thing.
Yeah. And like, you can't just Google, like, what a coral looks like. You're gonna get a lot of things that aren't what it actually is.
Yeah, and so if I try to, like,
You're limited to the literature, which is limited.
yeah.
Those whip corals, the ones that are just long lines, those are also a type of black coral. Yeah, they're not Elasella. Skeptical? What do you think they are? I mean, I can see the polyps. Ding, ding, ding. That's not how the octocorals look. I don't know. I don't see the chat right now. No, I don't have it open.
Do we have any chat?
Yes, we do. We have a question saying, any chance of seeing sharks being only 100-ish meters down? Thoughts?
There's always a chance to see a shark.
Yeah, it happens.
That is the answer that I personally like to hear.
It is relatively rare in my experience, but it happens. I have seen sharks before. When you get into the deep waters, you get, what are they called, six-gill sharks? These really ancient six-gill sharks.
Yeah, what about them?
They're usually really deep.
Yeah, deeper than here.
Yeah, but up here we might see some of the more shallow shark species. That's right. There's a researcher who's interested in some hammerhead species around in this area. So we're on our lookout here for hammerheads.
I see on the chat, they don't know what I mean, but when I said Skittles, Skittles is a disease that affects shallow water stony corals. It stands for stony coral tissue loss disease.
Yeah.
Skittled.
Someone in the comments, are they really only at 178 meters? Only?
Well, it's true. A lot of ROV dives are thousands of meters. But we are actually using an ROV to do relatively shallow work. Yeah, so this is an environment that's really not quite accessible by scuba. So scuba divers, yes, there are technical divers that can go this deep, but it's very extreme. So this is kind of this intermediate zone that is deeper than traditional work gets done by ROV teams, but shallower than is accessible by traditional scuba approaches. So we're kind of excited about using the ROV in this somewhat unconventional way. Certainly unconventional for me. I haven't done anything like this before.
I see someone in the chat asking what a black coral is. Black coral is a group of corals that make a proteinaceous skeleton. The skeleton's black, and historically people have used it to make black jewelry, like around from the Mediterranean, I think, and also around Hawaii.
So is it that the skeleton is black?
The skeleton's black, yeah. It's made of protein as opposed to calcium carbonate, like reef-building corals, chitin, or yeah, I think it's chitinous. Done with the 3D model?
I think we are wrapping up, yeah. So we're going to do some zooming in.
Oh, what's that stuff on it? Is that gunk? Oh, it's little deformities.
Oh, wow. This is a close-up of that Caligorgia.
How do we take a snapshot? Oh, frame grab. Oh, you just press.
Taller than me?
No, I don't think so.
No way. It's huge.
That's
It's amazing.
beautiful.
Yeah.
I think the stream was just buffery for a little bit, but it should be okay now.
So Sam, what do you know about growth rates? How old do you think that guy is?
This colony is probably a few decades old.
Oh, okay. So that's not as old as I might have guessed.
Yeah, these ones grow pretty fast. This is kind of also a pretty fast-growing coral, so you also tend to see a lot of damage, broken branches. Kind of grows fast and brittle.
Cool.
There's another species of Caligorgia that's a lot deeper, around like 400 meters to 800 meters, that we've taken pictures of every year for a few years to be able to measure its growth rate. And it can grow like about an inch or two every year, and that's at 400. That was at like
That's pretty fast.
400 to 600 meters. Those colonies. So this one's a lot shallower. Probably grows a lot faster.
Cool. Are you gonna sit down? Oh, you are. Okay. Alex is standing by with a squeezer. Oh, you're on the point.
Well, no, you're right. Sam, are we doing the Solaris? Yeah.
They're gonna. Oh. So we're getting ready to use this instrument called the Solaris, which measures superoxide concentrations. We're going to first measure the level of superoxide, kind of the background seawater. Then we're going to measure levels of superoxide really close to this coral colony here in a few places.
Yeah, so superoxide is what's known as a reactive oxygen species, and it's created just through the process of respiration, breathing, and it can be harmful in high concentrations, high quantities, but also it's a signaling molecule and can be important in all kinds of other physiological function of animals like corals and humans, and that's why why it's healthy for us to eat lots of antioxidants, right? chocolate and blueberries and things like that. Keeps our levels in check. But it's a very short-lived species. It reacts super fast, hence the name reactive oxygen species. So we can't take a water sample and bring it back up to the surface and measure it. So we had to build an analyzer, a sensor, and that's what this is here, that can make that measurement in situ.
Yeah, I'm doing it right now. Do they start it?
Right now they're just going to be positioning the wand, so they're not going to. They're getting set up for it. So first you'll start by measuring background seawater and see what the levels are away from the coral, and then we'll move in closer and tighter and see how the levels of superoxide increase generally is what we see, although not universally. And this is an instrument that we should note was newly developed. Colleen Hansel's group with myself and Jason Caput worked to build this, and William Pardis and Lena Tenser. And we first deployed this in 2019 off the coast of California. Made some of the first deep sea measurements of superoxide from deep sea environments. So this is really the second time that we're able to make these kinds of measurements. That's a worm. Yeah.
Would you guys be keeping this as the same event 3, or would this be a new event? Are you guys keeping this as the same event 3, Lena? Yeah.
I was yawning.
Yeah.
I didn't want to yawn and be rude. Even though there was no Solaris, there was no signal? Okay, because you got the eDNA sample? Yeah. Yeah. Yeah, so I think I was mentioning to our live viewers that we've done this, we've used this instrument once before on one expedition before, several different dives out on the west coast, and made some of those first measurements of superoxide from these deep sea organisms, including these corals like this. And so it's exciting to be back now, 3-4 years later, making some of the second measurements. You gonna hang that squeezer on the front, or can you place it back in the basket?
Well, we're gonna place it back. Whatever you want us to do.
No, I don't care. Whatever's easiest for you guys to get it out of the way.
Probably put it back in if we're not
Okay.
using it that way. We're gonna put that thing out in front.
Oh, good call.
Maybe we can get that one.
Tucked behind its ear. Oh, Lena, I just noticed the camera on the on Solaris pumps. It's pretty cool. So now Alex is going to put this squeezer sampler back into the basket. That is some crisp new polypropylene. That's a great question. One that our colleague over here, Colleen Hansel, should answer. Maybe she could turn on the microphone and answer that. Oh, it's not? I gotta answer it. Okay, so reactive oxygen species. I'm gonna speak intelligently about Colleen's research. So reactive oxygen species are definitely produced through photochemistry. In the sunlit surface ocean, that is a major pathway of formation of superoxide and other reactive oxygen species. So the question was whether or not the ROV lights could be creating levels of superoxide here through just abiotic reactions, photochemical reactions. And I think we have done some tests, but I wouldn't say exhaustive tests on making measurements and then turning the lights off on the ROV to see if that changes, and have not seen any effects. But it's something we should probably keep repeating. When we see signals, we should then turn the lights off and just monitor and see whether we... Yeah, exactly. Thank you. Well done. You can drop that. That's fine. Oh, I thought my stomach was telling me that it was dinner time. In fact, it's 5:30 on the nose. My stomach is growling online on the live feed. It's okay.
Let me know when you want dinner.
Yeah, no, it'll happen. It'll happen. This is too cool right now. Yeah.
Yeah.
There is a live feed of the dive stream up in the mess. You can still watch while you're eating dinner.
And narrate?
I don't think you'll be able to narrate, but
Oh.
unfortunately.
Nobody will want that.
For subsampling,
Yeah,
I can show you the size of the piece we'd probably need. Hand size. Yeah, it's going to depend a little bit on, this one has a little less tissue than others, so yeah, about a hand size,
5 and 6, correct?
maybe a little more.
like a Sam hand size, or different people's. Yeah.
Right. Our
Colleen's hand.
what are the size of the quivers that are on right now? Are they all?
Yes, we should
All of
have
them are?
concrete
Okay.
metric indices exactly.
Yeah.
Yeah.
Each of those sheets would work for five corals.
The way that lunch is, you're going to want to bring it like back towards us. Like in between. You won't notice that much. You're going to want to put it like, try to get it over here.
Yeah, I think our stream connection is a lot more stable at the moment. Huge thank you to our AVIT engineer for helping us out with that. We were switching over to another satellite there. That's why it was a bit buffery. And it is also, in fact, raining. So hopefully the connection should be better. And we really appreciate your patience.
Yeah, you can go and quiver.
Do we have a sense for how long this dive will be?
I think we're planning on pulling up a little bit around midnight.
Excellent.
Maybe a little earlier, maybe a little later.
Yeah.
Okay.
Soft, you think? That looks great. That looks great. This coral also, it'll be easy to snap it off if it doesn't cut. Yeah.
Well done.
And we usually put it face down into the quiver.
First
We usually
biology
flip
collection.
to go into the quiver.
Oh, after the sargassum. Is this one that's going to be incubated?
Incubated. What do you mean?
Like
Experiment?
kept alive?
No, not this one. Which pore size? 0.22? Is it the first of the 0.22s? Is the second of the 0.22s? And the first one hit full 70? How long did it take to do 60 liters? So it might be like 5 minutes or so to... I thought Luke wanted to... I think if we... Once they plug it, you can start it up again.
I'm curious as to what shows up as, or what might be expected to.
Yeah, that's a great question. Sorry that you missed the excitement. There actually, we were surprised there was no measurement of external superoxide there. So we've seen that before with some species of corals don't produce or aren't net producers of superoxide. So this was the first time measuring this species, and it seems, at least from the sample size of one, that the species was not producing superoxide. So, and we'll be continuing to do that later on. Yeah, it was basically the levels were the same as background seawater. So.
When y'all took Solaris out the first time, did you find anything cool with the instrument?
Oh yeah, so most of the deep sea corals that we measured, and sponges, and other kinds of organisms, were all very large producers of superoxide.
Cool.
In that case, and Lena Tenser here is working on a paper right now for those results. It was actually surprising. Only the black coral that we measured was not a producer of superoxide, and all these other octocorals and sponges and anemones and other cnidarians and things, actually, yeah, lots of organisms seem to produce it, which was surprising in some ways. Hard corals? Yeah, so this had a student that graduated has done a lot of work on shallow water. Are you talking about deep sea? So shallow waters in sunlit coral reefs and things. There's an instrument in the lab here called DISCO that is the diver operated in situ chemiluminescent analyzer or sensor. DISCO. Anyway, it's basically a shallow water version, diver operated version of this, and we've used it or Colleen and her students have used this in shallow water corals, shallow water settings, and there's all kinds. Oh yeah, no, and the scleractinians, yes, they are producers, but not all species. So Colleen would tell you, Perites, for example, is a very big producer, while some of these other species are not, and it's really not known why. But it's an active question that I think her group is working on.
So you just want to do a collection here?
Yeah, I just throw it in the quiver. I think we're going to swing around the basic, so the big thing is being on our way. Yeah.
And the other thing I'm gonna ask, so there's like 7 right here.
Yeah.
So it doesn't matter which one, but there may be one that's closer. There, what do I do? Zoom in. We're gonna see if it's fine. Yeah, some different species. Is it okay to like spend a little bit of time and just grab everything?
This looks like probably a Tanacetatapathes. A bottle brush black coral.
Where do you see the worm?
So if you
Can you just look at that real quick?
the middle, like right where it was focus.
Yeah.
Okay. But yeah, if you can select this one.
Retractable.
Oh, okay.
This is Stylopathes.
Currently having a bit of a watch change up here on the pilot seat. We have J-Rod as lead pilot, and now we have Dougie as co-pilot. We're just switching out a bit for dinner.
I can wait. Are you hungry? Jeremy, are they about to collect that black coral? You think it's a Tylopathes? That's very different from a Tanacetatapathes, right? Is that very distantly related to a Tanacetatapathes? Okay. Looks like that's some of the Tanacetatapathes we get in the Gulf.
Trying to chase you around. Basal plate kind of pretty flat, and usually top them off whatever is attached to it. Get the basal plate and the whole stem. I'm giving this one a base number 578. So if we create you don't do Solaris on this one, then we're skipping number. Or maybe you could just write 578 on there and X out Solaris. We're not doing it. Yeah. Put on the stuff. 578. That base number. That takes 18578. Okay.
Mark with an observation for the little coral next to like the one I just marked.
The yellow one?
Yeah.
Okay.
Do we know what that kind of yellowy one is next to?
The one with a little bit of green is a black coral. Like right here in the bottom middle. Oh, it's just gone. Well, the one that's right near the black coral is an octocoral. It's kind of orangey yellow. And there's a slightly green yellow one that's a black coral.
Yeah, because I already logged the one on the right.
The dark colored one?
Yeah. So what would be the other one?
Okay, to get a closer look, it's an octocoral. Probably a Paramuricid. We got that on there. It's probably here.
Are all anthozoans octocorals?
No. All octocorals are anthozoans, though. Because the hexacorals are scleractinians and black coral.
Oh, so the scleractinian and the hexacoral?
Ooh, nice. Plucked it.
It's okay. It's okay. Don't worry.
Okay.
You did your best.
Anthosauria, octocorals.
Yeah, those are those are black corals. Those are hexacorals, not octocorals.
Oh, so these are hexacorals?
Mhm.
Those are the octocorals?
Yeah. What? Is that quiver 2?
Let's look at the area. Pick something from that blue grass. One that has a distinct tube look. There's one right over here. That's just after this.
Whoa, blurropathies.
Blurropathies. Indeed.
Hmm? Yeah, we'll be collecting some octocorals. Oh.
Yeah. This is definitely a Stylopathes. And the tube, yeah, it's a perfect one.
Jeremy says stylopathies. Not sure if you can hear him.
Is it branch? Not branch?
No, it's
Not branch?
single branch, but it's pinnulated and subpinnulated.
Right.
Right. Oh, that's dense.
It's hard to see from here because there's so much tissue and subpigmentation. Let me try to fix
Oh, is there a worm in there?
Oh, but
There will be a worm.
is it because
Like right the tube there. Like you can see the branches forming a channel.
is it pinnulated but not branched because like the black part is in the middle?
So pinnules are a special type of branches. So if they're like you can see the tube there.
Yeah.
If they're of equal distances and spaced at equal lengths, they're like very regular branches. We call them pinnules. Same as like terrestrial like plant terminology.
Yeah. Ooh, that's nice.
That's good. Awesome.
I said a nice.
Yes.
Lisa said hi. What is this one, Jeremy?
Stylopathic.
Again?
Yeah. You gotta let them pump them all. There's so much here. Is there other
I don't think
Did we lose that first sample? Or we put 2 in that quiver.
I think Jeremy's putting everything that he wants in that one quiver.
So 2 went in there. Jeremy, is that 2 corals in that second quiver?
Yeah, we're definitely gonna change the name of that first one. I should have seen it. Did you want to collect anything else here?
Yeah, I guess.
Do you want to collect much?
Yeah, that's true. That's true.
Any of the soft colors, I think.
Yeah. Yeah, let's try to get that. The one that we measured, especially the one that we measured. We want to branch to the one we measured.
Totally different, and it's interesting for you.
Here, look at this, Sophia. As we've been collecting the sample. and like a typical Caligorgia, they got a lot of dead branches. Yeah, it just has a lot of branches broken off and bare skeleton and hydroids growing on it.
Calibration with an optical.
Yeah, primnoid. The one we measured, yeah. And then I think we got to have a plan. I guess we move on, try to find better ones to sample with Solaris. The colony. You're saying I control the camera? Ah, okay. Jeremy, can you do the logging then for this sample? Is that a cable? Is that attached or is it wireless? Oh. Is this the camera you guys use to see when you're taking a sample?
Generally, yeah. Right, left, right, forward, down, back, up, and then spin slowly to the right, and then
Zoom in. Okay. reach it from that far? Okay, well, let's go ahead. Did you guys get a frame grab? This is an Celid. No, it's a piece that can fit in a quiver, but probably we should probably do the largest piece we can fit in a quiver. I don't have the time. Quiver 3, does that work for you guys? Yeah, it should be 580, I think. No, this is not 580. This is 577. It's the one we measured earlier. It's not. It doesn't show up on the basket sheet because we didn't take a physical sample. But if you look at the Excel sheet on the this coral is going to be somewhat brittle, so it'll be easy to snap a piece off. It's not going to be one of those ones where the blade really needs to be in there to cut it. It'll come off. It'll snap off pretty readily. Yeah. Pretty densely branched, hard to get the hand in there.
Sam, which species is this again?
This is, we don't know the species, but it's an Celid. That should be, yeah, that's good.
An octocoral?
Oh. Is the blade on the right side of the claw right now? So is it gonna, is the branch that we want gonna fall off? Is the blade on the left side? Oh, I see. The red rubber will pinch. Oh, okay, okay, I see. No, I mean it's okay if you catch the extra branches in there, because it'll just snap. It's not like others where it just starts pulling.
Well done, Doug.
It should be pretty brittle. It should. I think if you rotate the wrist, it should snap at a certain point.
Wow.
Oh, it's pretty tough, guy.
A Nicella species.
Nicella?
Nicella?
That's what I was thinking I could be, yeah.
That's Andrea Quattrini.
Oh,
yeah.
Andrea's saying it's Nicella.
Yeah.
Write it down.
Write it down, then she knows. So yeah, we're sampling a deep sea coral here.
I guess we got more in there than
It's dinner time on Falcor 2, so we're got a lot of shifting people running around going to get dinner. But
you can open it back up. We've tried this
chief
before. We
scientist
open just
Colleen
a
Hansel
hair,
stepped out
so
for
that
dinner.
some of the then pinch back down. Usually that can help snap it right away.
Sam Vosen's directing traffic here now. So the technique here for sampling these corals involves grabbing them with a little blade on the manipulator and this branch of coral here is not coming off very easily.
It's more flexible than these guys usually are. Maybe now it's making ROS.
Yeah, maybe there's some superoxide being made now after it's experiencing a little bit of stress. This is probably something they encounter all the time, right?
Yeah.
Grazing. This is grazing pressure.
Can you continue? How many times around can the wrist rotate? Can you try like a one full rotation? Is it snapping or is it still tugging? Is that it? Is some of it snap?
Wow, this is. Yeah, we are using a cutter on this jaw. Yeah, do we have the cutter on the wrong blade on the wrong side? No. Okay.
It's just a weird coral.
Okay.
Oh, there we go.
There it came. Excellent.
They roughed up.
Nice patience there, Doug. Yeah, I've heard, stories from folks like Chuck and whatever who, back in the old days, they didn't have the techniques or whatever worked out how to sample these things, and often it would result in pulling up the whole thing and
the whole colony.
So
this one should be a sinker, but given its surface area and just how delicate it is, it could catch the current and go, but it's not buoyant. Not too buoyant. Yeah, you just open up a little bit. Yeah. banked. You got the record of it, Jeremy? Right? Cool. Okay. Then Colleen said.
Yeah. So I think next.
Caligorgia.
Yeah, I think we wanted to try to reposition and get into position for a Solaris measurement on the next Calagorgia that we see here in the neighborhood.
Okay, do you want to go east of that?
I actually don't know which direction is. Let's see. I'm taking a. Are those out there, Calagorgia?
Yeah, those big ones are.
Is that what are we? Oh, we're looking south.
We're looking northwest.
Oh, we're looking north. Oh, that's right. Okay, that's the front.
Did Santiago want to? What was the?
Eventually, we want to
dive
transit east,
No.
right?
Or east? Okay.
I think. Colleen says east.
Is it towards that waypoint on the map? Is that where we're heading towards, or the plan?
I'm not sure what that waypoint is. Oh, you can zoom. Yeah, zoom out.
Here's the overall map right there.
So we're gonna.
Oh, I see.
That's from the last dive. So eventually, we're gonna work our way east along the ridge. Yeah, like that. Add onto that nose. Yep,
Okay.
exactly. Not that far, but yeah, add onto the nose. So let's maybe start facing east and move along the ridge and look for the next Calagorgia. Yeah, that's what Andrea said.
I'm sorry, I've sort of almost abandoned this camera.
Abandon the what?
This camera, the controls of this camera.
Which camera is that, Luke?
It controls that one.
The 4K?
Yeah. So I guess I don't want to be.
Yeah. Yeah, and if you could just sort of, I guess, give us eyes kind of up and down periodically.
Do you know how the yeah, here you go.
That was a nice little pocket there.
It's like some kind of corals. Oh, is that in the lower right?
Yeah.
That's a Calagorgia.
Yeah.
Some.
And right below us there. Either one of these. Yeah, what is... Oh yeah, there's a bunch down here. Yeah, do you want to set down in here? We could pick one of these guys. Luke, Sam, you can confirm these are Caligorgia? Or which one is?
The one in the upper left is. That one is. This one in the middle does not look like Caligorgia.
Is it possible to be able to sit down to access a couple of those different ones? Okay. Let's try to do that.
Yeah, the one on the left is Caligorgia.
Yeah, that one looks.
Yeah, looks like.
Yeah, and if we could set up and maybe be able to also measure the two to the right.
Basket star. The basket star
on that. Yeah.
Ooh. Oh, what, Nicole? You taking a?
Sorry.
Take a video of yourself?
What?
Yeah, I think it might be 3 different corals, or at least 2 different ones. The big guys. Did you want a 3D model of some of these? Or are we going to see how that Caligorgia turned out? Oh, that's a good idea. Right, because that one.
Yeah, we didn't have to go very far, did we? To find these guys.
Right. Yeah, but we are also interested in the ones that don't make a lot of ROS, because that's going to help us understand why some do. Ooh, there's a dead skeleton up on the top left. Scott, do you see this checklist?
This?
Yeah. This is what we're doing to keep track of what we've been doing for each coral colony. Yeah, so now we'll be on the 5th row here for this new Caligorgia.
Okay.
And the SH number for this would be 580. SH18 580. And then we just been like marking which things we've done, and if we did a superoxide, we would write the event number for it.
Okay.
Is that an arrow crab on there? Oh, there's a big arrow crab on that. Yes, this is another Caligorgia.
Just taking a peek here.
Ooh,
at this guy.
That might be the Acanthogorgia that Jeremy wants. Ooh, take a frame grab at that.
That's I can't take credit for that. My eagle eye there. That was Andrea who suggested.
Oh.
Zoom in on this.
It might be Acanthogorgia. I mean, not Acanthogorgia, Acanthopathes.
All right. Do we have? Oh, we got Lena. Lena, how do you feel about setting up for a Solaris measurement here? Okay. So we're going to be using the port manipulator there. Extreme sour. All right, so the way this works is we're gonna have a background seawater. Okay. So Doug, this is gonna be Caligorgia. Yep. Just sort of want to get a second one to confirm. So this will be event 1, is that right? All right. Right now we're going to do a background seawater measurement, so just sort of have it off. I think so, yeah. Lena, is this a good position for the wand for the background seawater? Okay, let's lock that in there, and she's going to... I'm just, while we're taking this background seawater measurement, I'm going to take some close-ups of some of the... But I just got in close. Oh, yeah.
There's a bunch of arrow crabs on the colony. Has event 1 started yet, or just about?
All right, we're positioned right at the tip there.
Whoa.
Oh no.
Screen goofing up. This screen now is goofing up. Those are cool opioids.
Sure. What? Did I do anything? No? Yeah, the one on the left. Yeah. Okay. Do you want me to do another background seawater measurement before that? Okay. So Doug, I'm gonna zoom out here. You see the colony just to the left down there? Yeah, I think that's the purple one, gray one. That Colleen wants to measure that guy. Oop. That's reachable by you, right? Yeah. Yep. You ready to do that? Do you want? Okay. Let's see. Okay. We don't have a species ID on this one.
We can call it, I think it might be a Canthopathes. So
you think it's a black coral?
Oh.
Or no, Pathes, yeah.
Yeah, it's definitely a black coral.
Okay.
You can do observation, yeah. I thought you already did this one. Right. Or what'd you write? Acanthopathes. What were you doing?
All right,
Let's pull
Doug.
that for you.
Yeah, we can now move the wand in a little closer to this guy.
I think it was the same one.
What event is this? What event? Oh yeah.
PH, no I, right there.
Whose camera is that? Why does it look so much different on here than over there? Okay, now I see. Okay, yeah, monitors.
Could
Is this
be.
the one you wanted to do? Yeah.
Nothing. Jeremy eating? Which event is going on right now? 3? It's a black coral.
It is a black coral, yeah.
And we think it might be Acanthopathes. Or Jeremy will come soon and tell us it's not.
Is this going to have an SH number?
Yes, SH will end in 581. SH 18581.
Yep. Okay, we're done measuring with that, Doug. You can pull the wand. You want us to sample? There's some other species. Oh no! The whole arm went down. Yeah, it is. I mean, it didn't get hit by anything. It just, I think it was that the tip of the wand there just sort of got. you know, one thing we can try to confirm, I mean, the piece of tubing that's left on there may still be functional, right? Doesn't have the, was there a filter tip on the front? So it doesn't have, no, I know. So we're missing that now. But it's still, if we have flow, if we can confirm that we have flow there in some way, do you see any, are you pumping at all? Yeah, I just sort of want to know. I guess I just kind of want to know whether it's functional at all, because the bit of 1/8" plastic tubing that's coming off there could be still used as a manipulator tip, you know, wand tip if we need to. Yeah, well, and then I think you'll know, right, whether you're pulling in background seawater or not, right? Does it look normal? You understand?
That bullying harassment video.
I say we yeah, I think so. I think we limp through.
You know, it'll be a little harder to see where we're at, but it doesn't have a filter then, which means we have more prone to get a clog in there, but you can try it.
That's what I was going to say, exactly. We'll grab the other piece, yeah, throw it in a bio box. Yeah, right.
We've done sampling here, right? Or at least Solaris.
Well, there's two other species just to the right.
Oh, okay.
If you wanted to,
Okay.
that I don't know if we could reach them from right here. We'd have to jiggle to the side.
We might want to collect some coral samples. We measured white.
The Calagorgia, yeah.
Oh no, you could go ahead and grab that.
Jeremy, when I was doing C-Log for the samples earlier, I made it and I was waiting to click
that's
go
over here,
until
sorry.
it banked it, but it still used the timestamp from when I first hit the button. So I was wrong about that.
They're just down here off
Then
screen
you
to the
press
right.
the
See
button
over
and
there?
start making it.
Okay, do we know what it is?
Okay.
No,
Is that another category?
maybe. It looks like it's not quite as white.
Talking about the coral?
Yeah,
That's something other than Calagorgia.
right.
Okay, maybe we should try that one next.
Okay. Is the
You want to collect a sample, right?
I'd like the ones we measured if we could. Yeah, especially the black coral.
I think they're just going to pick up the wand right now.
Yeah,
The one. Okay. What's the
the tip
what's your-
that dropped off.
I saw that. Yeah, slid off. What are you thinking about that? Is that Solaris still functional? on the
Yeah,
side?
it seems to be. I mean, from Lena's assessment, that it's still pulling in sample and measuring. So we'll just use the piece of tubing there as the tip, which isn't so bad. Far more challenging things have arisen being undersea.
Yeah. You see that dead coral skeleton?
I did. When we landed here, I was trying to zoom in on it, and I did a terrible job.
So we have a few folks asking what just happened. So the telemetry dropped out on the ROV, on the arm, correction. But luckily, I'm also hearing that the Solaris is still working because the signal seems to be the same as
Yep.
it was before.
Yep.
Excellent. So not all is lost, thankfully.
No. These things happen. You're like, oh no! Oh no!
Libraries, and the very end of just how we're wanting it to be wild. I sucked it up. And then as I was moving it, I just started pressing down. And as it was dripping. And then
Is Jen
I couldn't
still around? No, Jen's gone.
You're on like autopilot. Like, no!
my brain was like,
Should we eat?
Jeremy, you want to take my spot? You know it, all this. I signed numbers already.
Oh, sorry. That's great. Good news. Solaris calibration worked okay, fine too.
Oh, good, okay.
Yeah, I think we just did. We'll just, what do you want me to call it?
Yeah.
All right. I don't know what path is. I like it. Hi everybody. I'm taking Sam's place for the moment while he has dinner. My name is Jeremy Horowitz. Yeah. I'm a postdoctoral researcher at the Smithsonian Institution. Hi everybody. Okay. Is that Dr. Jeremy Horowitz over there? This is Dr. Jeremy Horowitz at the Smithsonian Institution. So I'm taking Sam's place while he has lunch. It's not my watch, but there are black corals on the screen, so I ran. Dinner, by the way, not lunch. I still feel like it's 10 a.m. I swear. I went to sleep. I woke up and I was like, so yeah, having an eventful dive, aren't we? Yeah. Somebody at dinner looked at me when I went up there and said, "Man, you guys just hit the ground running when you got down there." We did. We really did. And then we hit the Solaris. Yeah, we hit the ground running, and then we stumbled a bit. Hi, everybody. Yeah. Our constant viewers. Somebody is online asking. Yes, we are we're on the boat controlling a remote vehicle. It is down the water, so we are not underwater ourselves, as in a submarine. There are other scientific vehicles like that, but this is a remote vehicle. Oh. I couldn't see over your chair that you answered that already.
Thank you for reiterating, though. That is a very commonly asked question. Yeah.
Okay, so we're going to pick up the wand. Are we putting it in one of the bio boxes? Yeah, we're going to have to put it in one of the bio boxes. Yeah, the reason why they all know me is because I'm as active as they are when I'm not on the ship. I'm on every dive, commenting, asking for looks on black corals. I'm a Falker superfan. Just Falcor? Falcor 2 superfan. Oh, you mean compared to the other vessels? Yeah. I think I'm partial to the Falcor. I hear you. Yeah.
Totally not biased.
I know.
it's nice to have the stream on in the background when you're like working or studying. Yeah, it's really relaxing. Especially like a 12-hour dive, you just like have it on all day.
There's just a ton of all kinds of stuff. There's a little zoo in there.
What are all the things on the bottom?
Yeah, it's like a little zoo down there. Like there's just covered, literally.
Well, there's coralline encrusting algae.
Yeah.
Like we're really deep, but there's still sunlight shining through. So that's what all that color on the floor is.
The red and then there's some of the yellow are sponges.
There's sponges. There's lots of a lot of these are cup corals, like
Oh, yeah.
scleractinian cup corals.
Saw those too.
Which are also of interest. Not, I think, in this exact moment, but on the expedition we'll try to collect some cup corals.
Sample collected.
I was ready to document a sample being collected. So in addition to narrating, we also need to document what gets observed, what gets collected, where it gets put. And when the Solaris is used, what coral Solaris is testing. Which coral are we subsampling?
We did that. Oh, you mean are we actually? Okay, yeah, we're gonna collect the calibration, yeah, for Sam.
Hey, Mom. Yeah.
I know one of my daughters is online as well.
I think my nephew is also online. Hey, Simon.
Hi, Kinsey.
Actually, it's my grandfather's birthday in a few days, and I named a species after him recently.
Of black
You
coral?
can't name it after yourself. That would be bad form. You can name it after...
What type of coral was it?
It was a black coral.
Oh,
It's called Hexapathes baikovskii.
nice.
It was described at the same time as Antipathes falcorei
Mhm.
from Falcor Original
Yes.
from 2020. That was a good trip.
Do you happen to have like a picture of it?
I do.
Okay.
I'll
Can
send
you
it to
send
you.
it to me?
Yeah.
Amazing.
Well, you may have seen J-Rod has a picture of a black coral on a nautilus shell.
Yeah, it's on the computer screen upstairs.
Yeah, so that's Hexapathes baikovskii. Have you seen that? It's on a nautilus
I
shell.
have. Yeah, that's a beautiful
Yeah.
photo.
Yeah.
Okay.
Those are spider crabs.
Arrow crabs? Are they spider?
Spider crabs.
Okay.
Okay. So we're collecting a Caligorgia, soft coral. Solaris tested it, but didn't get a reading.
That's right.
We're still waiting for our first positive reading. We'll get it.
I'm curious, are the coral cutters sharpened?
Yeah, they're very sharp.
Okay. Oh no!
It's okay, there's plenty more coral.
Some current. The opioid. Yeah. That's the joystick that came in. Twist. Yeah.
Maybe in the middle, there's some branches that seem unoccupied.
Like that one.
Yeah.
Is it far? What about that?
I mean, yeah, maybe not to disturb that.
About halfway between the end and that. You're right. A little bit, but yeah, they're right there.
So we're going to collect this colony, and then there's another coral just to the left of it that we're also going to collect. And then we're going to move on.
We may try to scooch over to the right of this colony. There's a few other colonies right behind it that I want you to tell us what they are, and then maybe we try to make another Solaris measurement with those. Yeah.
Sweet. So it's hard to know. Well, there was a question about how long it takes for corals to grow. Often in the deep, they grow more slowly than shallow water species. But for most species, we don't really know how fast they grow. But it's a large colony, so it's probably pretty old.
There's a question in here about the maximum possible depth for the ROV. The ROV is rated down to 4,500 meters, so very, very deep. I believe the deepest we went on the last expedition was 4,300-ish.
Wow.
So,
Yeah.
yeah, we can go pretty deep. It takes about 4 hours to get down to that depth, so
And with a rating of 4,500 meters, you've got access to something like 97 or something % of the ocean.
yeah,
Yeah.
yeah.
So it's always a delicate operation getting a fragment of coral in a quiver. The coral's slightly wider than the quiver. It's a delicate operation. We got Chris here.
Chris directing traffic.
He's a veteran. I think we need like Schmidt Ocean Institute baseball cards, but like
Oh, for
so
ROV
it's like ROV pilots,
crew.
yeah,
Yeah.
stewardesses, like, and then make them like playing cards.
Yeah.
Also.
Or like Pokemon cards.
Yeah.
Yeah, trade.
The captain.
That would be like a very highly valued one. There's only a few of those in circulation.
Totally. And then the scientists,
Also the Juliana card, very valuable.
and then like a ton of scientist cards.
Yeah, right.
Like,
Joe Schmo scientist.
totally. So it often takes time to.
Nice. Oh,
Yep. Yep. It's been collected. 18584.
yeah.
18584. And it's in quiver 4. This is a Caligorgio. It's a soft coral.
Well done. Well done.
Thank you for that, Doug.
How do you feel about some Solaris measurements on the species next that are nearby? Oh, you wanted to grab that.
So the Acanthopathes, that's what Sam's calling it.
Right.
Um,
To the left of this. Yeah. You want to collect it?
I mean, you already had it down to collect.
Right.
So, I mean, I don't want it, but since you said so. You're right. It's all recorded. I really want it.
All been documented.
Really want it. So if there is sargassum on this surface. Yeah, so it often takes about 20 minutes-ish to collect a sample. So although we're down for about 12 hours at a time, you work as hard as you can, and we try to get between 15 and 40 colonies, and of course do all the other experiments, which is why we're here in the first place.
Nice.
So some of these corals can float after they've been
Okay.
put in the quiver, so it's important to get that top down. We've lost specimens before, just see them float away in the currents.
Interesting. Yeah. And the cores just go burping gas the whole way. Yeah. Yeah, same thing, Jeremy, can happen with methane-rich sediment cores when you take them with an ROV. You come up and they start expanding, hydrate starts expanding inside, it turns into gas. Poof.
It sends off like a rocket.
Yep.
Oh, they have some good ideas. They said they need a merch line for World War II.
Okay. Looks like we've closed the coral quiver. Yep, looks good. I think we're gonna move slightly closer towards the next coral that we're gonna collect. So the yeah.
Totally looks like that's like a word cloud.
That's awesome. Is it Acanthopathys? We need to zoom when you get closer. And we'll need some nice stills before we collect it.
Okay. Yeah.
Certainly. Certainly not. You're asking if it's Caligorgia or not?
No.
I'm going to say no. And Sam is coming to confirm. Not to confirm, but he's coming and can confirm.
Yeah, are all those little white colonies out there, little hydrocorals? Yeah.
Mm. So, yeah. Okay, so it does look like Acanthopathys. Can you you're a bit far, right? Mm.
Yeah,
Yep.
you're right.
Yeah. Can you zoom in a bit more? Just look at the polyps. Yeah, it's beautiful.
We should. Yeah. Andrea's also requesting a zoom before we leave this area. A couple zooms.
Can we take screenshots? I don't have a. Someone at a computer to take a screenshot, a screen grab.
Want me to grab it?
Yeah, please. Just a few nice images. So what's cool about this coral is that it belongs to the Epanopathidae. However, it's over 200 million years divergent from the Epanopathidae. So we're going to need, and it's not related to any other species by over 100 million years. So we're going to be creating a new family for it. but we need really good molecular data, which is why we're going to collect the sample and we'll extract and sequence the DNA. So luckily we found one. Can we zoom out a little to get like a full image? Yeah. And then just like another screenshot. Thank you. All right, let's grab this beauty. How big is it? It's pretty small, right?
Bio box?
Do we have room? Is it okay to put it in a bio box?
I don't know.
Oh.
Colleen, is this a bio box? Can this go in a bio box, or is that not?
Okay.
You could put it in that same bio box, right? With the wand. That's okay with you?
Totally.
Is it okay to put it into the plastic bio box with another piece of plastic in it? Yes, of course.
Let me write this down. So we collected a lot of really cool black corals today. I'm really excited.
Okay, I just wasn't sure if you were watching.
Are you recording the sample collection, Jeremy, or do you want me to do it?
Yes, this computer's dead, so please. No, I don't.
Okay.
Thank you. Yeah, I think that's what I have. Yep.
No, wait. Hold on. Is it? Oh, you're right. Yes, that is 51. Yes, sorry.
No, we want the associates. It's ACANTHP OPATHES. Nice. And if we can collect the base as well, I know it's hard, but like if you try to like wiggle it side to side, the plate of the bottom of the plate will pop off whatever it's connected to. So just do your best. No pressure. Kind of like wiggling a tooth out of a socket. And yeah, it's also really important to know what associates live on these species. That was it. Perfect. So we got the basal plate, which you can see at the bottom. Beautiful. Can take a screenshot. Sorry. If we could take one nice screenshot of it, like just being held up, like that. Great. So the reason, yep, so that's perfect. So the reason why at times it's important to collect the full colony is because, well, one, a lot of the informative features that are used to identify a species are at the base of the colony. And two, when we think a specimen is really important and is going to be used to update the taxonomy of a group, it's important to have the complete colony So when future specimens are observed, all the different parts of the colony could be compared to what we call a voucher specimen, which is what this is.
Cool.
And that'll allow us to identify, confirm that other specimens are of the same species as this one, which allows us to understand the range of these species. And even collecting the associates on the species allow us to know what species associate with the species that we collected. So it's all about having as much biodiversity information as possible to be able to make informed conservation decisions. We're putting it in A, 1A. Oh, B. I made a mistake. That was
Yep.
Thank you. Awesome. That's a great sample. So this sample will be sent to the Smithsonian Institution, where it will stay there forever.
Awesome. And we got to see it collected. Set it free. Andrea, yes, was a zoom on some of the other.
Says before you move from the spot, you can also get zooms of the two big corals on the right of the
Calico, yeah,
The one up this way.
better if I
yeah, move, yeah. Yeah, to the right of the Caligorgia. She's also talking about the whips, but there are two species to the right over here.
That's not right. That's what I was
Yeah.
Not these.
No, I think she, well,
We can zoom.
right of the California.
Right.
So I was supposed to that right.
Yep. And we could get a zoom on the whips before we left here, if you want.
She said, and quick zooms of the whips too.
Oh well, too late. Sorry. That's fine.
So I think she must be talking about these guys.
Exactly. Yeah, I think that's it. And those whips are right there too.
I think she's talking about just getting a zoom in on these guys, and then also these whips.
So Andrea Cortriti wanted to be on this expedition, but wasn't able to go, which allowed me to go on this expedition.
All right. We got
So
Jeremy.
thank you for your sacrifice.
Yeah. And that's the stalked crinoid there. Yeah.
There are some folks asking in the chat about the Discord. Do you want to talk about the Discord?
Stalked crinoid.
What about it?
What's the group name on Discord?
Oh, well, I think there's a few people on there who can give that information.
Okay.
I don't know offhand.
Sounds good.
Yeah.
The opioid?
Yep.
Mm. Yeah, that was from earlier. I gave a shout out. Which one?
Maybe we turn autofocus off.
Maybe. Hmm.
Oh yeah, look at that.
I need to look closer. But we're not. I mean, it's. There's so much here. It's in this schizopathidae. Yeah, and then these are more of the stylopathids that have worms in them.
Oh yeah.
There's so much here.
The bottle brush.
We could stay here for weeks.
Yeah.
Is Andrea getting her zooms? Is she?
Is it on auto though? The brightness? No. I think that's a scleractinian. Little cup coral. And then just, oops, sorry.
Yeah, there's so much here.
Amazing. This is all just within like a square meter.
Yeah, there's.
This? The snail. The snail moved a little bit. Where's the polychaete? Oh.
So it's a bit of an overload of excitement
Yeah,
among
straight
all
in the
the
middle.
people here. There's too much to look at. We should probably move to a less exciting site, calm people down a bit.
Yeah.
Do we have any idea what the snail would be? Any
Oh.
idea on the snail?
No.
I think gastropod.
That was better than my no.
Says the geochemist.
It's a good guess.
Oh yeah, that's why we came
That's a
here.
black coral. Yeah.
And then what about the other?
Yeah, the one uphill from it. Whoa. Is that a basket star? Yeah. Basket star up there on it.
Wow.
Are you testing ROS? No.
No?
You don't want to just like wave the wand around all the corals in the area? Or just like...
I mean, it would be good to sample this one. We did one of the, if this is Decella, we got,
And you might as well touch the basket star. We've gotten signals off of them before. Yeah.
what do you think, Lena? Measure the speck? This big fuzzy white one? Yeah, sure.
See all this? All of it. Just all of it. Your watch is over there, by the way. I thought maybe you were giving it to me as a gift. No. Okay. We're not there yet. We'll get there. Sam, I'm going to pass this back off to you. Got my own shift to do this on later. I'll see you all later.
We have a bit of a watch change going on here. Got some new narrators coming onto the stream. Right now it is about 7:22 p.m. ship time, or on your screen it's in UTC 23:22. I guess I'll pass it over to our new narrators to introduce themselves.
Hi everybody, my name is Nicole and I'm from Lehigh University. I'm in the same lab as Sam, who's about to join us in a second. And I am here to help collect corals, identify corals, take some good images of corals, and also help sample some eDNA. And I'm a PhD student at Lehigh.
Can you hear me?
Yes, I can hear you. Sam, do you want to introduce yourself again for any new viewers that have joined us?
Yeah, anyone who's come on recently, my name's Sam Bozen. I'm a postdoctoral researcher at Lehigh University in the same lab that Nicole's in. She's the other person on the headset. And I study coral microbiomes. So on this cruise, we're going to be collecting some coral samples. And one of the many things we plan to do with these coral samples is investigate which microbes are associated with them to see if how they're related to ROS production in these corals. See if they contribute to ROS production or other things, like if the coral might use ROS to control which microbes associate with them.
So I think we're gonna start to try and take a ROS measurement here. And there was a request to collect that pretty large snail that we saw earlier.
Lena, is this event 1? Okay. This is Nicella. It's an elicellid. It's octocoral. Nicella, Nicella.
Yeah, I think that clump in front of it, that scraggly clump, that looks like a sponge. Or right. Where's the stick? Oh, that is a sponge. But I think keep going to the right corner there. Yeah.
Oh yeah. Those might be lace corals. They might be little stylastrids. A sponge.
I think we can call this a demo sponge.
Yeah. Is that something you guys would want to collect? Right.
Do we know what kind of differences we're expecting in ROS readings, like of the sponge compared to the corals?
I don't. That's something I think Scott and Colleen might know.
I'd be very interested to find out.
Yeah, they're discussing something right now, and we'll forward that question.
This event 5.
Are you seeing anything planned?
Oh, yeah, there's just leftovers. Most of the stuff has been put away.
Someone in the chat is asking what all those squiggly things are on the sponge. Oh,
Wow. You know, those
Nothing?
might be a type of like small tube worm. But maybe maybe tracks of like a circulid worm. If you're talking about the dark brown squigglies on the sponge.
Oh, on the sponge?
Yeah. It also looks like there might be a hydroid on one of the sponges that's kind of like blowing towards the screen.
Alright, I think we're good. Thank you. Okay. Well, good to see that there was a response. Alright.
Colleen, do you expect different ROS measurements between the sponges and the corals?
In the past, we saw that, like the corals, most sponges make peroxide, but not all. So again, like the numbers specific. Just never know. So it looks like that one had a little bit of a signal, but not a huge signal. But
There was something.
there is something. That's good. Our sensors work.
Yeah, that's good. Alright, so looks like we're gonna go ahead and take a little sample of this coral.
I can't quite tell if it's got short arms, so it wouldn't be too difficult. Yeah, I mean, we can always, well,
Does the species have the same hardness as Caligorgia?
Some Nicella seem to be as brittle and snap like
Oh.
Caligorgia does.
There you go.
This one just seems a little tougher. I don't know, maybe we'll have luck with this sample. There it goes. That's what I was expecting from the first one.
That's a nice cry. You can get all that in the fourth liver. Pretty impressed.
We're in a good spot for the snail.
Well, the black coral right in front of us, to the right of the Nicella we just sampled. I don't think we. Did you guys measure? I don't think you guys measured it. Did you? You try it with the Solaris?
Do we know what it is? What kind?
Big bushy black coral.
That's better than I could do. Bushy coral.
Sam, there's a couple different lip corals here. So I've seen these ones are a little more straight before in the Gulf, but these more like pigtail ones, are these different species?
I think the pigtail ones are probably a black coral. There's things called stichopathes that kind of look like that. There's a few others. The ones that don't coil and are kind of irregularly shaped, like the one right in front of the ROV, those I think are the octocorals. I think that's Elicella.
Okay.
Yes, that's the coral.
Yeah, that's a big bushy black coral.
That's Bushapathes.
Oh, we still got our snail friend. He's still here.
Colleen, this will be SH number that ends in 583. Are you guys, Lena and Erica, are you guys recording that kind of information, like the SH number associated with the, just like the species? Okay. You might want to write, what? What? Oh, she's writing down the event number?
Yep, I'm putting that into the C log along with the flow number.
Oh, okay.
It should be going in.
Oh, okay. I've been putting the random Solaris things. Are you pressing like the Solaris button?
Yes. I think when I wasn't here that didn't happen.
Oh.
So, but I got everything written down here. I got the event numbers written on here. So I think we'll be able to.
Oh, okay.
What event number is this going to be? Did you restart? Because Scott has one here again. Yeah, new file. Okay. Every time there's a new file, it data gets dense. Okay. All right. Yeah. Because otherwise I'm going to have to keep writing down the file.
Yeah. So when I read them over here, I guess I'll be telling you guys too. It's long, but it ends in, whoa. And those are just going to go up in numbers. So I might just say the last three numbers just to make it simpler to communicate. Colleen, when you were measuring ROS from corals on that previous cruise, did you measure any that have like whip growth forms?
No.
Okay,
That
because
would be really hard to do.
okay, that's what I thought. Okay, that's what I thought.
Yeah. I think it would be nice to start moving a little shallower, like where we can maybe get to some Switzia. So it's like I was going to move east and then start coming up a little bit. So maybe after we do this one, we can then start transiting.
Yeah, maybe fly, and while flying, if we see a coral that looks like it's worth sticking the nozzle in its face, then we stop.
Sorry, what's that? What's that? No. What? No, we were moving. We were still moving that.
Hello. Collected maybe, I think 7 or 8 corals. We had Caligorgia, bottle brush. Jeremy got another second coral that he put in there. Octocorals and black corals. Yeah, but we've been focusing. We did 2 Solaris measurements and samples of Caligorgia and Nicella. We're checking. We haven't really got a signal from any of the corals. Stuck it in a sponge osculum and got a little bit of a reading. The arm lost power, went limp, and hit Solaris on the rock. Andrea said she wants that snail. I said Andrea said she wanted that snail.
She mentioned it might be a good slurp target. I don't know how big that is. If the slurp could handle that.
Well, it might be a little bit big because at the end where the stuff usually enters the canister, it's like this narrow. I think
yeah, it's a narrow.
Snail might be able to blast the ROV team. This one? Wow, I don't know. That's huge. Yeah, it's at least an Elicellid, we know. We think it's Nicella. Yeah, we were thinking Nicella, and then Andrea came in and suggested Nicella. We don't know if it's the same exact colony, so we just moved on and found another Caligorgia. I figured it might take a while to try to find the exact same Caligorgia.
Do you want to sample this coral?
I guess we should, since we Solarised it. Yeah. Yeah, we want a piece since we did Solaris. It's part of our diversity element of that of this project.
And while we're sampling, I got our lovely PI on a headset. Colleen, could you introduce yourself, please, in just a few seconds?
Sure. Hello, everybody. My name is Colleen Hansel. I'm a senior scientist at Woods Hole Oceanographic Institution, and I'm the chief scientist for this cruise. My team has developed the instrument Solaris that you're seeing, the wand that we're putting up next to the corals. And what we're trying to do is measure reactive oxygen species, which is you can think of it as a really reactive form of oxygen. And the hard thing about measuring this reactive oxygen is that it has a really short lifetime. So it lasts for about 30 seconds in the ocean. And because of that, we know very little about where it forms, how it impacts the ecology and the biogeochemistry of the ocean, and in particular in the deep sea. So a few years ago, we developed Solaris, and this sensor has been out on Alvin and made some measurements of deep sea corals for the first time, but we only had a limited amount of time to make some measurements, and it was really exciting. What we saw was that some corals make these reactive oxygen species outside their cells, and some, or outside their colonies, and then some corals didn't. Same with sponges, some do and some don't. But what we want to do coming back here with Schmidt Ocean Institute was follow up on that study and really dive into looking at a lot of different species of corals and sponges, and now in collaboration with a team of biologists and ecologists to really try to understand why this is happening, why these organisms are making these really reactive chemicals in the environment.
Yeah. That's an amazing explanation. My question for you is, and a few folks in the chat have been asking, is what helped you decide to come to Puerto Rico to study these corals? What makes them special, and what interests you about these corals, specifically in terms of reactive oxygen species?
Yeah, so
Puerto Rico is really interesting in the sense that we have some really beautiful mesophotic corals, so corals that are below kind of most light. There's still a little bit of light in the upper mesophotic, and then we transition down into really low light levels, and then also we can go into the deep sea corals that live in the absence of light. So we have this wonderful transect of corals that are living in very, very different environments. Also, Puerto Rico has pretty low productivity. So what I mean by that is there's not a lot of really reactive carbon, really bioavailable carbon in the water. So these corals are really having to work to live and to make a living in these waters. And so we think that maybe the production of these reactive oxygen species that can break down carbon are helping them live and essentially helping them acquire food. So it's a great place for us to be doing this research.
And I guess just for the folks listening back home, could you give us a sense for some of the projects and research groups on board this specific cruise?
Sure, we have a great diverse team, people from different institutions, different backgrounds. So we have people from Woods Hole Oceanographic Institution, from Lehigh University, from the Smithsonian, as well as the University of Puerto Rico. And we have chemists and geochemists, biologists, ecologists, deep sea biologists, microbiologists, so just a wonderful diversity of people here to work together to explore these deep sea animals and also their microbiome. So what are the bacteria that live associated with these corals and sponges, and then how are they playing into some of these chemical transformations that we see. We have people also that are studying sponges as well, so looking at the production of these reactive oxygen species by sponges, and then how that relates to some of the ways that these sponges change nutrients in these ocean environments. So just a diversity of different scientific disciplines that we're all kind of coming together to try to collaborate to make some headway in understanding this process. Oh, I should also say that we have engineers here as well. So Solaris was the result of a team of scientists and engineers at WHOI that I led. And this was a really challenging sensor to make because of the fact that this reactive oxygen species, superoxide, the one we're focusing on right now, has a lifetime of 30 seconds. So what we need to do is, typically in oceanography, you'll collect waters, you bring them back on board onto the ship, and then you measure them in the lab. And we don't have that option because by the time we get it back on board, it's gone. So we had to put, the sensors into the sea to be able to measure it. The tricky part is that we have to kind of do chemistry in the ocean. The way we measure superoxide is we pull water into the sensor. It reacts with a chemical probe that produces light. And then we measure that light in real time. And so because we're changing through different light environments, different pressure environments, different temperature environments as we go deeper and deeper into the ocean, that impacts then chemical reactions. They slow down as things get colder. So we have to actually calibrate then in situ as well. So we have to make our own superoxide in the instrument and then be able to essentially standardize it. So whatever signal we're getting, we know exactly what concentration we're looking at. So it was a very challenging endeavor, and luckily I have some wonderful engineering colleagues and friends who were up for the task, and it took us a few years to build Solaris and make it, and it's been working wonderfully. So we're very lucky to have this wonderful team all working together.
I'd be interested to know in some of the challenges y'all had while developing Solaris.
Oh, there was a lot of challenges. So,
Yeah, so one of the trickiest things I would say in building Solaris is that these reactive oxygen species, as the name implies, are highly reactive. So we couldn't have any components within the instrument where it would have some sort of reactive substrate that would degrade the superoxide. So no metal, no plastics that aren't inert. So any wetted part where the water sample was going to come into the instrument, that had to be completely inert material. So that was really challenging because a lot of the pumps and things that we needed, we had to build in-house. So at Hui, we were developing flow cells, developing different pumping kind of, I guess, pumps, I should say, and check valves and things like that, and that had no metal in it or no reactive forms of plastic. So that was really challenging. The first time we actually have a shallow version of Solaris called Disco, and the first time we built Disco, we put it in the water, and we weren't getting any measurements at all, and we thought, oh well, there's no superoxide here, and then we realized that there was a really tiny piece of metal in one of the check valves within the instrument that was scrubbing it all. So I would say that was the most challenging part. Yeah, where did the name come from? Going to try to make me remember what it stands for. It is an acronym. I tend to like making acronyms, and so like the DISCO is the diver operated submersible chemiluminescent sensor. So we cheated and put an O, we took the O from sensor, turned it into DISCO. Solaris is a submersible oceanic luminescent analyzer of reactive intermediate species. I remembered, all right. So we liked the Solaris as a name too, because in the past we really knew nothing about these reactive oxygen species within the ocean, but we always thought that they were really constrained by light. We know that these chemicals form when you take carbon and it gets illuminated by sunlight in the surface of the ocean. And so for a very long time, no one thought that these reactive oxygen species could form in the dark. And so when this was discovered not too long ago, in the last decade or so, that there are dark reactions that form these same compounds, we thought it was kind of ironic to call it Solaris, which, for the sun, since we're looking at it in the absence of the sun. Oh, and there we have a sponge. Is that a glass sponge? Do you want to measure the sponge? Okay. I think we're going to measure the sponge. Do we know what coral that is there with the basket stars on?
Yeah, I think it's a trick.
Tricked you.
Yeah, my friend, he thinks that orange coral is Paramecium.
Would that coral be worth looking at as well?
I think so. It's nice. The other big one that we haven't measured one that's closely related to that one just yet. So I
Okay.
think it's worthwhile. It's like a different family than the ones we measured earlier.
Thank you.
I'm more interested in measuring corals on demo sponges rather than glass sponges.
Is this a glass sponge,
Yeah.
you think? You could try it and see what we get. Yeah. Do we want to try the glass sponge?
I'm not a sponge expert.
Yeah. Do first measure on the outside and then go on the inside? What event are we looking at? Okay.
Yeah, Steve's confirming what we said, that the coral could be Paramaricia, or at least a Paramariciid. Steve says so, yeah. Steve Auskovich.
Yes. So we'll try to go outside first, and then
He's on the chat.
just
Yeah.
on the outside, maybe near the surface of the sponge, but outside
Oh,
of it,
yeah.
and then we'll move inside. That's probably good. What do you think about that location? You can see it.
Is very interested in sponges and Sunday could be associated with them, and she just walked into the man at the perfect time.
All right, I bet we can move over to the inside of the sponge now and see. Try, if it's possible, try to get the tip in as far inside of the center of the sponge as possible without touching it.
Anything from the sponge? Measuring it? Any signal? Kate, do you want a piece of that sponge? We're sniffing it. Only if it's making a lot of ROS.
Has it found anything?
We stuck it in a sponge earlier and got a little bit of a reading. That's it.
We're good.
Yeah.
Good, Lena. Yep. Yep. Oh, true. That I think we wanted to measure this this other coral in behind the wand right now. I think we're collecting background right now, so I think we can just go ahead and move right to it.
The coral? Wait, no, I'm asking the coral. You're asking? It's we think it's a It's hard to pronounce this. Paramericiid. The family Paramericidae. Could be Paramericea. Could be a bunch of other things. Don't know. Well, I don't know. Yeah, but there's lots that look just like it too. Like look up Placogorgia. Placo. 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 There you go. Gorgia. Type in Placogorgia rudis. Rudis. That looks similar. No, Placogorgia rudis. Placogorgia rudis. See? Pair of shoes. That looks pretty similar also.
I think your first guess was right.
A lot of them look really similar. Really hard to tell them apart, even when you.
Is everything pumping okay?
Unless you look at under the microscope.
Okay. I think we're good. What do you want to do? I think that would be great.
What are they saying? Calibrating it? A recalibration?
We're going to try a recalibration of the sensor since we're not getting any signal. We're a little confused. We typically see, we would expect to at least hit somebody here that's making superoxide, and we had a little bit of a signal earlier on one sponge, but none of the corals so far. So it's a little perplexing, but so we're gonna check the sensor and just make sure it's functioning properly. Yes.
Earlier you said that you thought Swiftia would have or produce a lot of ROS. Is that from a previous measurement or last dive?
Yes. So when we were did our first dive with Solaris, and we measured, I think, 5 or 6 different coral species. Swiftia's was one of the highest. So
like yesterday or on a different?
No, on the previous cruise in 2019 when we were on Alvin.
And
okay. Okay. Okay, so it seems like our sensor is working, but what we're finding is that the calibration standard is decaying really fast in these waters, which tells us that there's a lot of stuff in the water that's decaying this chemical, which means we may have a hard time measuring it unless it's really high. So that's okay. That tells us something. So.
Well, there's a little opioid on that thing.
I think
it looks like it could be.
I think we'll probably want to collect. Sam, do we want to collect a coral?
Yeah, please collect the yellow Paramaricea that we just sniffed. That's a black coral. Yeah, it's a small one.
The coral that we just tried to measure superoxide on this guy, they'll want to take a little piece of it so that we could characterize it. Yeah, so someone mentioned that maybe we're having a hard time making a measurement because of the current, and I think that could be playing into this. As you can see, the current is really high in these waters. So that could definitely be playing into this a little bit. The fact that we're having really high decay. Just makes it a little more challenging.
Is it usually abiotic or biotic factors that lead to a higher decay rate?
So the primary way that we think that superoxide decays in the environment is a reaction with abiotic factors like carbon, metals. There are microbes that will decay superoxide. There are extracellular enzymes that decay superoxide as well, but we believe that most of the decay pathways for superoxide is like reactions with things like iron, metals that are in the water, and carbon. So you can see a lot of particles flying around in these waters. So it could be that there's just a lot of, and there's a lot of biomass right here. So it could be that there's a lot of carbon in these waters. That's just degrading the superoxide.
Yeah, I wanted to ask you, in 2019 when you sampled Swiftia, was there this much of a diverse benthos with all the CCA?
No, so when so the first time we deployed Solaris,
and measured superoxide was in the Monterey Bay Marine Sanctuary, National Marine Sanctuary. Thank you. And it was on Davidson Seamount, which was a very pristine area and did not have a lot of coral cover like this. So it was a seamount. This benthic cover. Not a lot of coral and algae all over the ground. There's a lot here. We are deeper right now. Oh no, in Davidson we were a lot deeper. That's right. We were we got down to about 2,000 meters at one point. Also, the colonies were a lot bigger. But actually, that's not true. Swiftia was small. So it'll be interesting if we can find a Swiftia here, because we know that species makes a lot of this extracellular reactive oxygen species. So I would expect to see it here.
Unfortunately, I think there's been some revision of the things called Swiftia. So the Swiftia
we're looking
like
at Coral Quiver 6. So maybe we'll have a hard time finding that same species.
No, I mean, all the things that were called Swiftia aren't necessarily the closest relatives, and the Swiftia we have here, it's not quite like the others. So the one you guys sampled on that cruise, probably distantly related to this Swiftia.
Okay. Yeah, so we see this species variability throughout actually all the different organisms that we've tested for this process.
Quiver 6 already has something in it.
It does?
Can you please go to Quiver 7?
What is in Quiver 6?
The black coral, the bushy black coral that we sniffed a little bit ago.
Sorry, J-Red.
Seven's empty, 7's free, 7's good.
That's right. So yeah, someone was asking about the shallow coral. So we've done a lot of work in shallow corals with our other sensor, Disco, and it seems it doesn't matter the geographic location. So for instance, there's a common shallow coral called Perites. It's a very, very hardy coral, and it makes extremely high levels of these reactive oxygen species outside their cells. And we find that no matter where we measure it, and no matter what species it is, so within the Perites genus. So there's a lot that we don't know about this process. We just don't have enough measurements yet, and so this is going to at least help bring us a little closer to having more measurements. But we're seeing the same thing with the deep corals, that some make it and some don't. And we think that it's an essential part of their physiology. So at least we know for plants and animals, including humans, that we make these compounds to serve a lot of different processes within our bodies. And so that includes repairing wounds, making nutrients more bioavailable, fighting off pathogens, lots of different reasons. It's kind of the Swiss Army knife of chemical compounds. And so and what regulates when an organism is making it and when it's not. We know a lot about that in plants and animals and humans, but we know pretty much nothing about it in corals. So it's a fun area to start studying, but very perplexing at the same time. What's the name of this coral again?
A paramuricid, Paramuricid. It's the fam. It's like the family.
Okay.
Popular species studied in the Gulf of Mexico after the oil spill. A lot of these Camerosia, Skya, there's people have taken a lot of images over time to see how they've recovered.
Oh, very cool. Sam, is this 584?
Yes.
Wonderful.
Some corals can be more challenging than others to put in the quiver. Sorry, I think you heard a little bit of what I was trying to say, but those of you guys who are watching, I think some of you have watched many corals be put into quivers, but some of them are more challenging to collect than others, as some are neutrally buoyant, which is okay to put in the quiver, and some are positively buoyant, so maybe you get them all in the bottom of the quiver all nicely and then they will float away. I don't think we'll have that issue with this species.
Sometimes to catch those ones that float away, we fly the ROV up the second we let go of it to keep it in the quiver. Yeah, then plug it while we're flying. Colleen, what I was trying to say earlier about the Swiftia, I guess in simpler terms, is that the Swiftia from the Pacific is probably in a different family than the Swiftia we're going to have here. Like the taxonomy is going to be revised if it hasn't already.
Well, we'll see if they
Oh yeah.
similarly produce superoxide. I think we're gonna start transiting up a little bit. We'd like to get a little shallower. And then probably northeast. We're trying to move a little bit.
Hey Scott, what kind of sample do you want to use the squeezers on? Oh, I guess if we detect the ROS. Going shopping.
I mean, in a way, we are going shopping. That's what
Bone teeth.
that's what we call it.
Like that one we first measured.
Yeah, like things like this.
Mm. Okay, they can get inside and outside easily. Okay. The big, that's an osculum. The big osculum, okay. Can I see that guide?
Getting to some of those species that hopefully we've measured in the same families or whatever, but because we've never had it.
Oh,
Yeah. I mean, but we're at 170. Out of Chris.
Scott, I'm gonna put the guide here. I don't know where it was.
Yes. Along the ridge, but heading kind of up to shallower waters. Yep. Northeast. Yeah. See a fish.
Category, we're gonna be like, okay, whatever, move on. Or what was the other one I made? Nicella.
So
Or can't stand these.
There's a fish. Yeah, I mean, we could stay here for days. So exactly. So it'd be nice to just get moving a little bit.
Yeah, that looks like that black coral we already sniffed and didn't see anything. Can't quite tell.
You thinking that looks a little. I'm still training my coral identifications, but I don't know. Looked a little. Oh,
that looks like a different black coral.
This yellow one on the left.
Yeah, that's cool. That's probably like related to Aphanopathes. I'm sure Jeremy wants a piece of that.
What about this one?
I think we measured one like that already.
Okay.
And didn't smell anything. There's a Caligorgia. There's a lime green octocoral behind it. Oh yeah, this looks like a Aphanopathes. This yellowish black coral. I don't know if you think it's worth sniffing it because the black corals don't make anything, or so far.
Yeah, I think it would just be nice to get a little shallower for a little bit.
Yeah.
Yeah, we're at a depth of 170 meters currently.
It seems like a good speed to me. What do you guys think?
Yeah.
Yeah, covers, but be able to still see things.
I think things might get a little different when you get to maybe 100 or 80 meters.
Yeah, when we start to get more light, we'll start to see different things.
Yeah.
Where are you seeing the depth? It's not this here? Oh, okay. So that's not.
But yeah, it's also up there.
Oh yeah. That might be a different kind of Nicella. The little orange guy.
Oh, I see. Okay. But that's not. That's different than that. And that. Okay.
Ooh. Is that a little Paramecia?
Yeah, it looks like similar to the one we measured earlier. All these little orange guys look like they might be little Nicellas. That's like what we see in the Gulf. Oh, look at that pink and white one. No, it's too small. It might be a different kind of style asteroid.
What is this guy? This little pink guy here?
We think it's a style asteroid. Might be.
Yeah, there's some
on the
sponges.
sides. Oh, there's two demosponges. Oh wow.
Do we know what this thing is up here? This.
It's probably another style asteroid.
Yeah, the pink and yellow thing is probably a style asteroid.
Okay.
Did I say pink and yellow? Pink and white. pink and white.
I am in training when it comes to my coral IDs, so
I'm sorry.
I'm at least confident about this one.
I think we could keep going. Yeah. Yeah, is that a scleractinian?
No, but it's a hard coral, but not like a hard coral. It's not a scleractinian. It's a different
Just
class.
pseudo hard.
Yeah. It's calcium carbonate. I mean, you probably haven't measured them before, so it would be interesting to sniff them at some point.
Yeah, we're definitely seeing a lot more of those little sponges there.
Oh yeah.
Things definitely changed here pretty quick.
Somebody asked how dark it is at this depth. I think it's pretty dark at this depth, right, Sam? I think you start to lose a lot of ambient light around 70
Yeah.
meters, 70,
yeah.
80.
Depends on the area. It looks like the light it's clearer
far.
waters here because in the Gulf of Mexico, at this depth, there wouldn't be
Yeah.
any of the pink algae.
Oh, true, true, true.
What is our depth?
It's probably a little bit of
It is
ambient light.
159,
150.
160-ish meters.
What the heck is that?
What?
When you're using the
Oh.
Solaris, can you give us a sense for what's a big number, what's a little number? I personally have no idea what, like I can't orient myself to that, yeah.
What?
Yeah, so a lot of the
No.
times what we do is we think about it as times above background
Can we
seawater.
look at this column,
So-
this coral here? Can we get a good look at this one
Oh, okay.
head on?
J-Rod, could we take a look at which coral?
It's like up
Oh.
to the left, to the upper left, off the screen. It's a taller white one.
I think it's maybe this one over here. Oh.
Is it this one?
Yeah, this one I wanted
Yeah.
to see.
Oh, is this chrysogorgid looking thing?
I don't know what this is.
It's something that kind of tricks you and looks like a chrysogorgid far away. Because it has such small, delicate
Well, it's very
polyps.
different. It's a very different kind of octocoral.
Yeah.
Okay. That's good. That's what I wanted to see. Thanks. Still not quite sure what it is.
It's got big polyps.
There's another interesting one, but I'm sure Sam saw it and would have would have wanted to take a look at it.
This one here.
It's out of the screen, but that's interesting.
This thing in front of us looks like another Nicella,
Oh, I see.
or whatever that Elicella it is. This coral looks like another one of those ones I asked to get a zoom on.
Do we know what this is here?
I think that's another one of those Elicellas that yeah, it's like the thing we took 2 samples from. It's just a little bit more irregularly shaped.
Wow, that's
It's crazy looking.
large. Oh, you can see the current with that.
Yeah, it's leaning over.
Oh, okay.
And you can see the particles just flying by.
Oh, wow. Yeah, looks like little shooting stars.
You need to find a little nook
That's
to
really
hide in.
strong.
Yeah, Swifty in the Gulf kind of starts at about 100 meters, goes up to like 60.
But I thought we've seen, not we, but Swifty has been seen here.
Yeah, there was up shallower.
Was it shallower?
Yeah, up on the higher up on the ridge. There's a soft coral right here that might be. It's like mid of the screen, a little bit lower to lower. Yeah, do you see that? totally different kind of octocoral. That's a
Ooh.
that's a really soft coral.
Yeah.
Do we know what that one is called?
Oh, I don't know. There's something like it in the Gulf of Mexico called chironephthia. That's like some kind of nephthia, something. Not sure.
Should we try to measure this one, Lena? It's an octocoral. Yeah. Let's do it. Your current's going crazy.
Ouch.
You okay?
Yeah, I'm good.
Oh, that's pretty. Wow. This is just complete covered. There's just life everywhere. You getting the background seawater, Lena? Yep. What event are we on? 2? Do we know what to call this?
A coral? Just call it a soft coral for now.
Maybe someone in our audience can help us.
Steve mentions scleronopytha.
Scleronephthia.
Thank you.
Yeah, it's just difficult because there's a few from different families that look very similar to this, like Scleronephthia and chironephthia look like this, but they're from different families now. I'm looking at Andrea's tree.
This is a great angle. We can actually see how close we are. Oh, I see how polyp close.
Yeah, that looks like
Coral
they
did not like that.
get a little shy.
Oh, they're back open. Who's sucking on me? How long of a lag are you seeing, Lena, in the signal?
Holy cow.
Did you do that, J-Rod?
Colleen, this will be number 585.
Okay. No luck. I don't know what is going on here.
You see, look at these two. See, this kind of looks like that, where we're at right now. So this is just a bigger kind. But it's really soft like that. There's 2 different families.
Well, we good? Nothing? I think we're good. Did you want to collect
piece of this?
please. Let's collect a piece of this. Or actually, it's probably small enough to pluck it, put it in the quiver.
It
Yeah,
looks like
this
a
is a sponge. Yeah.
These, yeah, it's a bunch of them, huh?
Quiver 8 is open. Is that the one you guys are going for? You got,
On the water complex, in the pool, you see I know it's species specific, but that's going.
are you guys discussing switching it to measuring hydrogen peroxide? Solaris?
Could be a pivot, but we haven't spent a lot of time working on that method, so that would take a little time. At least on Solaris, we haven't. But I mean, I think Lena made the point that maybe, you know, it's nighttime. The corals may, be slowing down their metabolism. Maybe on a circadian rhythm. We see that in the shallow corals, and it may just be enough to plus the high decay, plus the high current. Maybe we're not seeing a lot right now, so maybe we'll see more during the day.
Is there like a cutoff point for like some kind of transition between corals that have a circadian rhythm and corals that don't in terms of depth?
So I think all. I mean, this is kind of outside of my expertise, but I believe that all animals have a circadian rhythm. So like we have a circadian rhythm, and so you don't have one. So I think it's just that in the light, there's more of an influence of light on that circadian rhythm, and then in the dark, animals still have a circadian rhythm, and it's still kind of this diel kind of change in their metabolism and their physiology as a function of like time of day, even though it's not light driven. So we typically, see metabolism slow down in the evening and then pick up, in the early morning. Oh, what's coming out of there? Oh, wow.
Oh, I thought I saw a little bit of that before.
Wow.
The quiver was filled with fresh water. Do you guys always fill that with fresh water beforehand, not seawater? Yeah, I think we'd rather have it be in seawater, just in case. Well, I guess it's gonna float out because it's fresh. Okay. We're gonna hopefully be able to do the opposite on the second leg when we get the RNA later. Yeah, and the RNA later will sit in there in the bottom and not mix too much. Hopefully.
I think it's pretty difficult to study circadian rhythm in deep sea corals just because it's hard to keep them and we don't know a lot about their genes and genomes. But a good thing to think about is blind cavefish. So they live in dark caves and well are blind, so they live in darkness and also don't have developed eyes, but they do have circadian rhythms, but they are a little less robust than their like shallower water surface cavefish.
Do you know what other factors influence those circadian rhythms?
That's a really great question.
We could go wake Anne up.
Yeah, this would be a great question for
This is
Anne.
Excellent. Okay, I'll ask her later.
Yeah.
Circle back.
I don't know, I'm wondering if it's just something that hasn't been lost. Or no, because I'm just thinking out loud. But deep sea corals would be older. Yeah. We'll ask Anne.
Exactly. Before we just start making stuff up.
Yeah.
Oh. Darn bungee.
What about what? The coral? No, it's not. It's not Isella. It's too flexible. I don't know. I need a closer look. Are you trying to open that quiver, or what's? Oh. Yeah, you think it'll fit in the quiver? If it'll fit in the quiver, then yeah. Yeah, this one won't. No, we've measured this one. We thought maybe since it was so different, if we measure, yeah. What do you think? You think we have a different strategy? Caligorgia and Isella.
What is this one? Okay.
Santiago, do you remember what depth you saw Swiftia in those videos? 120-ish. Okay.
They don't want to let go. Gotta do the little shimmy. Almost there.
There we go.
Yay! Where do you think we're at with reagents? Okay.
Can you tell us more about the reagents in the Solaris?
Tell you where they are?
Yeah, where they are and what they are, how they work.
Sure. Yeah, so Solaris actually consists of kind of two main parts of the sensor. So you're all seeing the wand,
And in the wand is it's kind of our analyzer. So that's where we have kind of a flow cell that the fluid coming in from the wand is mixing in that flow cell, and then there's a manifold in there where we mix in a reagent. That chemical probe I talked about earlier that specifically reacts with superoxide and gives off light. And the reagents are held in a little basket that we, or I guess it's not a basket, they're kind of IV bags that we hang on an assembly that is on the basket of the ROV. So we have these really long tubes that connect these reagents to this wand. And so the reason why we analyze at the wand is, again, because the superoxide has such a short lifetime that if we tried to pump all the way through the tubing to the basket on the ROV, again, the superoxide would be gone. So we have to do everything right up in that wand. So instead, we pump the reagent to the wand. So we have a bunch of different kind of reagents there. So we have the chemical probe that reacts with superoxide, gives off light. We also have an enzyme called superoxide dismutase, or SOD. It's an enzyme that all organisms make, our bodies make it, and we use it, and this enzyme specifically breaks down and scavenges superoxide when concentrations get too high in our body. So we use this enzyme to in the Solaris sensor so that if we're making a measurement, we're seeing really high levels of superoxide, we can then dope in a little of this enzyme, SOD, to show that the signal should go away. And that tells us and confirms that, yes, indeed, we were actually measuring superoxide. It's just a check, just to make sure that we're not getting some strange artifact. Which at this point, we've been doing this long enough that we have never witnessed an artifact, but we just want to be safe. And then we also have our calibration standards on the basket of the ROV in this reagent assembly, and these reagents are again pumped into the analyzer. So I mentioned earlier, we're essentially doing chemistry in the ocean, in the deep sea. So it's fun and challenging, but it requires kind of a lot of pumps, a lot of tubing, a lot of kind of crazy analyzers. And so that's kind of where the reagents are. So you don't see it because it's sitting on the basket, and it's actually kind of tucked up underneath SuBastian right now. But we have cameras on it so we can keep an eye on it just to make sure that things are okay in that little assembly. There's a lot of sponges here. A Swiftia?
I see a sponge that has like a big one single hole opening. Like one of the big ones that has a small opening at the top? Yeah, or any sort of like vertical, obvious, singular hole. That's something we're sort of interested in at some level.
Kind of the ones that look like tubes. Wow, those are some big sponges over there. I don't know.
I think they're mostly glass sponges. We're starting to get those hydrocorals.
Okay.
Some other sponges. Oh wow, look at that big white one all the way on the right.
Yeah, it's like a vase. That's crazy.
Looks like another little Paramerissia in the center. Oh wow. Oh, there's a good sponge with a big hole. There's a couple.
Little white one?
Yeah.
Oh,
Those are
wow.
crazy. Ooh, what's that
Oh,
purple thing?
Oh wow.
I think that's that purple one that I've showed you guys from some video a couple years ago. That was here.
Sure. Yeah, what do you think? What do you guys want to measure? Want to measure that? The big purple thing?
Purple? I want a piece of that.
You can tell that some of us are not biologists. The big, it looks like cabbage, red cabbage to me.
Oh, that's
Yeah.
exactly the color.
Oh, we think this might be a Mauricia. Might be called Mauricia. It's like one we have in the Gulf. It's a good thing.
Something different.
Yes, please.
It's hard to tell.
Oh, this place is so fun.
That sponge is crazy.
Some dancing hydrocorals.
Oh, there's a lionfish. Right there, at the bottom of the screen there. You could land on top of the lionfish.
Yeah, just, you know. Oops.
They are invasive, yeah.
I can't remember if it's a
Harpoon
lionfish. I think they have like 300 million like babies at once.
They have 300 million babies?
I thought it was it. I can't remember if I'm getting confused with
Maybe like eggs.
sunfish.
That
Yeah, like
sounds
300 million
terrifying.
eggs. Might be sunfish.
I mean, that's common, isn't it? A lot of fish make a ton.
Yeah,
but only a few survive.
that is a lot.
Wow, look at that.
Oh yeah.
Yeah, they have a lot of lionfish derbies in Florida. Asian carp? Asian carp? In the Great Lakes. Well, not to prevent them to getting to the Great Lakes. Yeah. Mm-hmm.
This is crazy cover where you don't even have a place to land. Oh, I bet.
Oh. Colleen, do you think we'll sample a couple things here?
I think we are. I think we're gonna look at that purple coral and perhaps a sponge or two.
Okay.
It's just trying to figure out where to sit without smushing something.
Right. Trying to decide if I want to take a suite of eDNA samples here.
Ah,
If we're gonna be here a bit, and
seems like that would be a good idea.
yeah, I think there's quite a bit here to keep us busy.
Yeah,
And a lot of interesting things to see if we can pick up with eDNA.
that sounds good. Yeah,
All right.
it's definitely different than where you took the last one, the last sample. So
Yeah.
that'll be a nice
Yeah.
comparison.
All right.
That's a beautiful shot. You need to do a little frame grab there. Oh, the ooh, look at that. It's so pretty.
polyps going in and out because of the
I don't know what to
current.
call this. We're gonna call this purple octa. It's either that family or the family of Plexauridae, one of those two families.
Are you chasing the lionfish with the wand? Are you ready, Lena? What event are we on, Lena? Got it. I'm just going to call it purple coral. Mauricia.
That's what I said. Who? Who? Yeah. What? Mauricia? No, Mauricia is in the family Plexauridae.
How do we spell Mauricia? For those
M-U-R-I-C-E-A.
Okay. Just like it sounds.
Well, actually, it's Santiago that said that could be Mauricia.
Oh.
And Andrew agreed. Yeah, good call. Are you ready for it to get?
Yep. Sam, is this 586?
Oh, dandlefish.
Yes, it is 586.
Oh yeah, look at the.
You picking anything up? You think that purple pigment would react with ROS?
It could.
What does your guys' readout look like? Oh. Oh.
We have a signal.
We found it. Okay, purple coral is now a target.
All right, now we like the purple coral.
Now we really like the purple coral. Okay, we found a target now to get more of.
So what makes this one different? All right, biologists, help me out here.
What do they say? Pain is beauty or something? The coral's got a beauty is pain. That's the cost of being colorful. You gotta deal with all the ROS or make it.
All right, Lena, you're just happy you want us to sit here for a while? Make up for the last few hours. Yeah. Well, we're definitely moving back and forth. So very interesting.
Ooh, cool.
Is this a hardy? Well, we don't actually know what the species. Oh, we do. Do we know?
Might be Mauricia.
Is this a hardy coral?
Ooh, I don't know. I don't know.
So what we see with shallow corals is that the coral species that tend to be really resilient to stress seem to be most tolerant to heat stress, to pathogens, to disease, tend to produce a lot of these reactive oxygen species, and you can think about it almost like an armor, that they're kind of chemical warfare. So we see that consistently with shallow corals. So that's why I was asking: is this a hardy or
we got a signal?
Has a strong resilience to stress? We got one, Santiago. So I was just asking: is this a hardy species? Do we know anything about why Mauricia would be different?
The species of Mauricia we have in the Gulf of Mexico has a pretty wide depth range. Well, we study like Swiftia and Mauricia there. Mauricia seems to have a wider depth range than Swiftia. I don't know, maybe can tolerate a wider range of conditions than
Okay.
Swiftia.
And one of the most abundant corals at these depths was Solaris.
Okay.
So maybe they have some sort of capability of.
We got a bushy black coral. We got
Okay.
some bushy black coral.
Coral work to keep alive in Mauricio?
No clue.
The outside adults. Are they?
I don't think so.
Okay. I think we can pull away now, J-Rod. Thank you. Yeah, that would be a great, yeah. That would be great. Can we move it so it's kind of in the same orientation? Oh, okay.
Yes, it's the biggest signal of the whole cruise.
We'll take it.
So was the background seawater at the beginning of the dive.
Oh, yeah. Did you log an eDNA in the? Yep.
Yes,
Okay,
yes.
I'll put that on here too.
eDNA is
I
pumping.
didn't record the time. It might have been like an hour ago or half an hour ago. Probably an hour.
Andrea was suggesting they might have photosynthetic symbionts, this species.
Yeah, pigments.
This one? Well, the octocorals, when they have symbiodinium, they're usually not a bright color like this. They're usually like a strange greenish brown. This would make me think not symbiotic because that wavelength of purple almost on pink is one of the wavelengths that chlorophyll absorbs. You ever see those grow lights with the LEDs that have just blue and pink? That's kind of close to that, a little darker though, but kind of close to that pink. So that means that it probably wouldn't be available for the chlorophyll.
What's this? What's the species that produces caffeine?
Some Paramerissia. I forgot which Paramerissia.
Oh, okay.
What did she say? Oh, mm-hmm. Yeah, so someone was asking if they had photosymbionts, would they make more ROS? So superoxide actually can't pass biological membranes. So any organisms living inside the coral, so inside the tissues of the coral, actually can't be contributing to the signal that we're measuring outside. So what's being produced is actually being produced outside. So this is kind of the coral itself that has enzymes that pass through their membranes, or it could be bacteria on the outside of the coral colony that's producing it outside the cell. So there's different compartments of where these reactive oxygen species are made inside cells. Sometimes they're made inside, sometimes they're made outside for totally different reasons. But with superoxide, they can't pass from one compartment to the other. So while the photosymbionts could be producing intracellular concentrations, we wouldn't be seeing that here. That's not to say that they couldn't be having some other indirect role in the signals that we're seeing, but not directly.
Yeah, I did. I saw that, yeah.
And someone also mentioned that the current's a little lower here. It does seem like the current's a little lower right now.
But notably, she's not saying anything on the upcurrent side of this species right now.
Are we the same distance away from the?
It's a little bit further away. He didn't know.
Oh yeah. Are you sampling, Lena? Yeah.
Maybe if we can do the experiment, get close, start pulling it to the left a little bit, and then
So there's a little bit of a lag on the instrument, so maybe we could do stops along the way and see. Anything, Lena? Huh. Sure. Interesting. Huh. Oh, that's interesting. Yeah. Yep. Yep. So yeah. So we're not going to see it if we're not on the right side of the coral with these really strong currents. All right. Well, should we do some sampling? You good, Lena? Yep. All right. Let's retract Solaris and.
If we have time.
Sam, what kind of piece are we getting here? Just something for the quiver?
Yes.
Okay.
Please.
So just a smaller piece.
We could image it if you think we have the time. If not, I completely understand.
Yeah, no, I think we could image it. Yep.
I know you kind of have some barriers, so maybe not so zoomed in. Whatever's easiest for you, because there's a lot around you. Yeah, that would be not ideal. Okay, sounds good. Oops, fish starting. Yes.
Okay.
Oh, okay. Yeah.
Okay.
How about just some images instead?
Yeah.
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 Okay.
You never know what we could do with what we already have.
Yes. That's an image, and then we can sample. That's okay. Let's, what do you think?
I think this could be. We do have some good video footage up close. I think, showing some of the polyps. Let's get, while we're sitting here really nice and steady. Let's look at some of these polyps. We have
Okay.
some good lighting. Okay, let's see. Yeah, it's definitely current. You could make a 10-hour YouTube video of just this. Okay. I think those are some good images. You can always pause and get a still from, ooh, yeah, steady for like 2 seconds there.
Yeah.
That was nice.
It's not holding still.
Really interesting. It's like purple in some spots, orange on another.
Okay. Yep.
Yeah, I think we have good images. Okay.
Should we fire a Niskin bottle here for some just geochemistry data for context? Yeah, we could definitely do a sponge. I don't know if you want to do these. These are more open if we want it. Yeah. Well, I think we did. We could cruise around a little bit. Oh yeah. Okay. We could put it in a bio box. Yeah. Yeah.
I was talking to Andrew.
Sam, I think we don't want to destroy this sponge by taking out the basket, so we could just put it in a bio box, right? Does it matter?
Yeah, it doesn't matter. The only consideration would be how many samples we plan to collect for the experiment. So every bio box compartment we take up, we probably wouldn't be able to use that for any, well, unless we use this species for an experiment too. But we're targeting Swift, yeah. Never mind. Yeah.
Yeah.
So we can still collect. If we put this in the bio box, then we'd probably be able to do 4 Swiftia for the experiment today.
Okay. So we can put, I guess, only one in each section of the bio boxes is what you're saying?
Yeah, I mean, if it was something other than the experiment, just collecting for genetics or something like that, then I think it'd be okay to put 2 different things in the same bio box or do it in species. But since we're saving them for the experiment, if one goes in with the black coral and the others by themselves, it might start fighting with the black coral, and that colony may be kind of goofy compared to the others for the experiments.
That could be a sponge that later you replicate.
We could collect the sponge, yeah.
After we treat it.
All right. Okay.
Are we 9?
Is quiver 9 is still open?
Yep.
That's fine. Yep, that's good.
Oh my god. Do it again. Okay. I was speaking, just in case you guys didn't hear me. I mentioned some 3D modeling. This might be part of a project to see if the surface area or the total volume of the corals contributes to their ROS measurements. This is something we haven't tried out yet before, but it seems I've helped do some 3D reconstructions of hydrothermal vents, and it's surprisingly very flexible to get good video that you can construct a 3D model of a coral with. But somebody asked about the softwares. We haven't tried out any softwares just yet, but there's a mix of open source and paid softwares, so things like Mushroom or AgSoft. This is something that's becoming a really up-and-coming hobby for a lot of people. There's a lot of YouTube videos and that walk you through step-by-step how to create little 3D models of anything you might be interested in. So hopefully this is something that will end up being informative later on in the cruise. Earlier, right when we got started during this dive, we took some images of Calagorgia. So hopefully I can
Same size we've been doing.
Hopefully I
big
can
little bit in the quiver.
test making a model of that tomorrow and see if there's any improvements that we can make. One of the difficulties I see is that, you know, these corals are very detailed. We want to get as much as the polyp as we can. So it will be a fun challenge.
If it's Mauricia, it's Plexoridae. Aren't in the same order as they are there. No, because we have these here, yeah. Yeah, or... Yeah.
Get off.
Yeah. Just leave a small piece for a voucher. I think you're right about the when to do this. Instead of banking it, it's probably better to do it when the coral's there so we can have the picture, because it takes the picture automatically when you press the button, right? Well, yeah, then and then just wait to confirm it until it's banked. Yeah, right. That's probably the best way, because so we get the picture, but also you don't.
There's a bunch of little critters swimming around. What are those? What are those little critters swimming around?
Oh, it's from the brine shrimp hatchery. It must have spilled.
Oh.
I don't know. Little crustaceans or something.
There's a bunch of them. I think one might have ended up in the Quiver 10. Gonna go for a ride. Oh. Yay. It's a tight squeeze in there.
So I think we're getting ready to deploy what's called a squeezer, which is a device that collects water, which we're going to use to measure hydrogen peroxide. Because we just got a positive signal of superoxide from this coral. It's the first coral that we've seen a good signal from, but that is a measurement of superoxide. That's what Solaris measures. We're going to measure hydrogen peroxide with a squeezer back up here on the ship.
What are your objectives in measuring the hydrogen peroxide using the squeezer? Like what does that mean?
Oh.
So hydrogen peroxide is another reactive oxygen species, but unlike superoxide, it can pass through biological membranes. So it can be coming from any part of the kind of zoo of kind of symbionts and organisms that are associated with coral. So bacteria, algae, or if they're present, it can pass through membranes, and so we can get this kind of composite signal of this reactive oxygen species. So hydrogen peroxide, you're all probably familiar with. You probably have a bottle of it in your medicine cabinet, and we use it because it cleans wounds. So it degrades or kills any sort of bacteria infections that you get in a wound, and it also helps kind of repair the tissue of your wound. And so just like that, corals and other organisms, we make hydrogen peroxide, and it's one of the main reasons is for wound repair. And it's also because it can pass through biological membranes, you can also get this signal that's inside cells, and that's a little bit more of a stress signal. So kind of looking at the two, we like to look at both superoxide and hydrogen peroxide, and it kind of gives us a better picture of kind of how these reactive oxygen species are being formed, and then whether or not they're kind of related to stress. Unlike superoxide has a lifetime of about 30 seconds, hydrogen peroxide is a little longer. It depends on how reactive the waters are around it, but typically it can last for several hours, sometimes up to a day or two. So what we're going to do with this squeezer is it's a kind of a syringe sampler where we can hold it near the coral, and then the manipulator will squeeze it and it'll pull water in if it's behaving properly. Sometimes they don't behave. And then what we'll get is just a water sample in the squeezer. Then we're going to bring it back up on the ship and we can measure it. In another sensor we have, the shallow, actually, the sensor we use called Disco. So we'll measure it back in the lab. Ideally, someday we would love Solaris just to be able to measure both of these together in situ. We just haven't been able to get to that point yet in our development and design.
That's really cool. I noticed when you were talking about hydrogen peroxide, it does come in a bottle that's dark I believe that's to prevent light decay. What differences do you see in hydrogen peroxide at shallower depths versus deeper depths, and does that affect what you're trying to measure?
Yeah, so exactly. So the dark keeps the light from decaying the hydrogen peroxide. So in surface waters, you get both faster production because peroxide can also form by reactions of light with organic matter, or also from phytoplankton that live in surface waters produce a lot of hydrogen peroxide. So you get more production in the light, but you also get more decay because of that light reaction. So there's a lot of abiotic reactions that form, or just pure chemical reactions that form these reactive oxygen species as well. So both of these chemicals, both superoxide and hydrogen peroxide, we think a lot about them as kind of fluxes because you have both production and decay happening at the same time. So sometimes, like before, when we were not seeing superoxide associated with these corals, some of the corals we were measuring doesn't actually mean they weren't producing. It could just mean that the decay is faster than the production rate. So some of the water that will be coming up in this squeezer sampler will also measure the decay rate of these reactive oxygen species within the water. That'll help us constrain more kind of what these production rates are.
Yeah, and we realized that we got to orient the sensor carefully with regards to the current to see the signal. We turned it to a different part of the colony, didn't see anything. When we went downstream, we could detect it again. So we're thinking, we need to be more careful when we measure other corals. Maybe we just missed it on some of the other species we were measuring.
I mean, they wanted me to try yesterday, they pointed it back on downstream, but also after months down here. The black coral was kind of
There's a black coral.
the current was crazy. What? Yeah, well, the swim in the plate, the bottom of the plate. That's good. Might be squeezed that way. Yeah, that plate. So I could see the.
It's got subpinnules.
So the squeezer acts like a piston. So you see the manipulator is holding the bottom, and there's kind of a release mechanism in there. If we squeeze it right in the right position, it should release the piston, and then it'll slowly pull in the water.
It looks like there's a piece of white plastic and it's the flesh holes again. I think you're right. I think each one of these is like the whole thing.
Yeah, Elisola has like paired polyps like this. What? Uh-oh. Trying to move after this?
So these squeezers have been giving us a little bit
I
of
don't think we're
trouble
probably
in the
not
lab.
gonna sample that black coral.
As you can all see,
We're
it's
gonna
just
try
hard
to
to
find some
manipulate
sponges and
kind
more
of
of these purple
instruments
ones.
down deep in the ocean,
Yeah, that is.
and these
What do you think it
squeezers,
is?
you know, we want it to pull the water really slowly, so that piston is, the seal of it is really tight. So sometimes it just doesn't fire. Scott, can you remind me who developed these squeezers?
Well, yeah, we, they chose them as pretty, my ancestor there developed by Mario.
Okay, we think these, we borrowed these squeezers from a colleague, Jeff Wheat, and we believe they were designed and manufactured by Embari out in California. So we thank our colleagues for lending these to us. They may be telling us that we're using them wrong. It's also challenging because you can see on the manipulator on this arm, we have coral cutters on this manipulator. So we had to take off what is normally kind of the end of this arm, which would be able to hold the squeezer better and be able to kind of more wrap around the whole squeezer. But since we're wanting to be able to cut pieces of coral, you know, we're kind of trying to do it all. And sometimes that means that we're kind of losing some of the utility of the manipulator. The other manipulator, the other arm of SuBastian is being dedicated to the Solaris wand. So we're kind of only working with one arm here. So we're really challenging these pilots.
There's another lionfish in the background.
Oh, there's a lionfish.
Looks like he's checking us out.
Checking out the squeezer.
He's a reactive oxygen species fan.
Do we want to try another squeezer? One of the different ones?
4 and 6 would be options. Yeah. Do that.
Uh-oh.
There you're gonna put just the regret it. Oh, right.
Don't question the pilot, Scott.
Very good.
We do have kids.
or 6? 6 would be great. Okay. Expired last time.
How big were they? Well, I mean, like the first three.
Scott, I think you're muted.
Better?
Yes.
Yep.
There you
Okay,
go.
there we go. Yeah. Yeah, that shouldn't be a factor. The way they're designed, there's no, they're all isobaric. They're all the same pressure on all the sides and inside.
Lucky number 6. Like that fish go across the screen. Fish is like, what are you doing? What are you doing? What are you doing? Yes.
Hmm? Yeah. Instead of the. So these squeezer samples were originally designed to sample diffuse hydrothermal vent fluids, which are sort of low temperature, sort of barely oozing out of the ground, And those are difficult to sample because most sampling techniques draw in water really fast. And when you would do that, you would see mostly background seawater. So these were designed as kind of slow, gentle samplers for those to slowly pull in water from a very specific location. So they're really ideally suited for the work that we're trying to do here with the sponges and the corals, but where we want to draw water that is more or less the water that's directly around the sponges or around the, or within the sponges or around the corals. So I'm pretty sure these have never been used for this purpose, which makes it kind of fun and exciting.
Scott, did you explain how the squeezer works? The squeezing mechanism works?
Sure. So what these are basically a spring-operated syringe. So you're familiar with a syringe. It has a barrel and a plunger, and that's the lower part there with the big glass syringe, a big titanium disc on the inside, some O-rings to keep it sealed. That's the syringe part pointing down. The upper part that the ROV is, the manipulator's got in its jaws, is a circular trigger mechanism that gets squeezed, and there's a spring that's under compression inside there, and a firing pin that allows the spring, once released, allows that spring to pull on that plunger. So again, as I mentioned, they were designed to be to pull slowly and gently. So there's not a ton of force in that spring, and there is some resistance on the glass and the O-ring, just by the nature of the contact of those two materials. And there's a check valve on the very front, and that combination there is a delicate balance of pulling and resistance, and we've been working out some of the challenges of that. So the first challenge is to get them triggered. The second is to have the syringe actually withdrawn up into the trigger. It's a slick mechanism. I mean, I didn't design it. I think it's pretty slick, and I think it's worked well in lots of previous applications. This is my first time at sea working with them, however, so.
Do you have an idea for how many times they've been used out at sea before?
I don't. I do know, for example, that I had a former postdoc named Carly Buchwald, who went on a cruise with Jeff Wheat to Dorado and brought these with them. There was an Alvin cruise. It was her first Alvin cruise, and she got to go down in Alvin, and they used these, and that was in 2014, 15, something like that. Okay. And I think they were established techniques back then. So yeah, I was just saying that the first challenge is getting them to trigger, but the second is you might trigger it, and the resistance on the barrel inside the glass might just be too much for the spring. So yeah, the intermeshing fingers on the claw is what they're designed for, and would probably confirm we're definitely triggering, but still may not fire. Yeah, so we have the longer springs in there to try to increase that force, but that one fired.
Nice.
Yay!
Yay, we got one. All right. Yeah, we're shooting 50-50.
Squeezer number 6. All right.
Yeah, actually, we could just stop now. Right? Quit while we're ahead. That went faster than I was expecting. And the sargassum outside is accumulating. Colleen.
I can show a little CCTV of that in a minute.
What, the sargassum?
Yeah.
Yeah. Well done, Alex. That was faster. Pulled faster than I thought. So then, since we did get a successful squeezer, and you're logging that, right, Colleen?
I'm putting in the first squeezer here.
That's number 6. Do we want?
I'm going to
let's
call
see.
it squeezer 1 though, because that was our first squeezer, and
Okay, but
that's
it's
squeezer number 6. I got it.
okay.
Do we have an idea of how long that took to fill?
Less than a minute.
Less than a minute.
Yeah. As in, like 2 seconds, 4 seconds. Yeah, it went faster than I thought it would. Do we? Do we want a Niskin? Is there a point to a Niskin here? Okay, as a corroborative or collaborative, a complementary sample to what's near the coral. I guess not. We'll save that.
We could collect it. I mean, because we'll get more geochemical data than we're going to be able to get out of the squeezer, yeah? Or what will be different?
Well, that's the question. I see it normally as a pair that's comparing at a site near the coral or near the sponge and not. And so, but the coral squeezers are dedicated
The
to
squeezers are going to do
and VOC.
and VOCs, right? So it's
Right.
actually not complementary. It would be
Different.
measuring a whole other suite of geochemistry information, which we could get. Like,
Sure.
Yep.
So that we could just, for this particular time and site, we can fire a Niskin.
yeah, I think it might be worth doing.
I'm still holding out high hopes for a pair of sponge squeezers. Yeah, is the oh, never mind. It's not important. I'm just wondering if there's a seabird on the sub. I know it can be up here somewhere. Oh, thanks. So in
No,
case,
I would love one if you wouldn't mind.
but if you want for your notes, what the water background for your notes, if you want to know what the temperature of the water is, or we just it is, but in case you just wanted, it's the depth, temperature, conductivity, salinity. Oh, there it goes. Dissolved oxygen somewhere. Yeah. And there's and there over there. Oxygen's 218. That's probably micromolar. All right. What's our status? We got that stowed, Arnie? Nice. Do you want to fire a Niskin? Or are you shift changing? Ask Colleen.
I think we're still in the general vicinity, right?
Yeah.
I think it's okay to just go ahead and we can fire it if that's.
Do you guys do that with buttons or with the manip? Okay. Got it.
Good morning. We've had
Ben's back here.
Ben's back. We're having a bit of a shift change here. Huge shout out to Alex for getting that wonderful fluid collection.
Yes, I'm good. That's my
Now
seat.
Mike is subbing in.
It's a nice fish there.
Just a quick update, I guess, for anybody tuning in. This is our RV Falcor 2, ROV SuBastian Livestream. We are, woohoo! We are in Desecheo Ridge, sampling some wonderful mesophotic corals. Also using a Solaris sampler to sample reactive oxygen species in and around the corals as well as the sponges. And the time right now on the ship is 10:15 p.m. Or on your screen you see 02:15, that's UTC time. And yeah, thank you all for joining us today. And if you have any questions for our deep sea experts, please drop them in the chat. Yeah, and we did have someone talk about the sargassum seaweed. I guess from a crew perspective, it really depends on, opinions vary a lot depending on who you talk to. I'm sure the engineers aren't very happy about the sargassum. But a biologist will probably tell you that it's an important carbon sink and is an important resource in the environment. So it really depends on who you're talking to.
One is not firing.
Sure.
Mm-hmm. There it goes.
All right, 2 fired. Just background site characterization, chemistry. Yes.
I just logged it. Sorry. Yeah.
See you, J-Rod.
Yes.
Hmm. Yeah, we're just traversing along the ridge. We were kind of looking to try to go shallow, right? Yeah, so now we're kind of southeast, I guess. We're kind of at the shallowest point, aren't we? Now. Yeah. East by south, east southeast. Yeah. I mean, let me just. Are we gonna? The plan is to kind of come along down this. Yeah. Is that what you're saying? Can you zoom out one level on that? Yeah. So this is the Nautilus area. So we're gonna head that way. Just looking at time, if you want to recover at midnight, it's gonna be. We're not gonna be able to do much over there unless we just boogie over there. Hmm. But it could be 2 a.m. too, or I don't know. If yeah. Everything always takes so much longer, yeah.
Scott, we have some new viewers tuning in. Could you explain a little bit what we're doing? Just a very general overview of our transit along the ridge.
Yeah, sure, I'd be happy to.
Thank you.
So we've been working today along the Desecheo Ridge in Puerto Rico, which sort of lies east-west between this small island, Desecheo Island, and the northwest corner of Puerto Rico. And we've been traversing along the top of this ridge, which had previously been investigated once by the EV Nautilus, maybe 10 years ago. Showed abundance of coral cover and sponge cover and biota at the top of the ridge. So we've come up onto the ridge this afternoon and have been traversing along here at depths between 180 and 130 meters, which really is sort of right at that the deepest extent of the mesophotic zone where light penetrates. So we're seeing this transition into mesophotic types of ecosystems here. Very abundant. You see here on the screen very high abundance of sponges, glass sponges, and corals. All kinds of even smaller scale stuff. When we get zoomed in on things on the benthos there, it's overwhelming how much is really there. You zoom in onto a small section of the benthos there, and it's like a little zoo, and there's just so much life. So there's, from at the macro scale here that you can just see how diverse and abundant the life is. And so we've been working with our sensor for reactive oxygen, it's called Solaris, to understand something more about the generation or not of this reactive oxygen species superoxide by these organisms here, and working to collect samples of them and samples of water to try to, again, understand that connection between the chemistry and the biology in these kinds of environments. So these environments are hard to study, virtually impossible to study by conventional scuba kind of approaches. So while 130 meters is pretty shallow for an ROV, this is really one of the best ways to get into this kind of environment. So it's pretty unique. I think I think I can say this is the shallowest I've ever worked with an ROV. And I was,
Same.
I was very excited about coming on this cruise for that reason. For
Mhm.
one of that being one of the reasons. So
Yeah.
thanks, Jeremy, Dr. Jeremy Horowitz. Everybody is in the house. Right. Yeah. Yeah. So when the ROV works in very shallow waters, the water is intrinsically warmer, and often there's currents and things that the RV is working against, which requires it to use its thrusters more, which puts off more heat, and so there can be overheating problems that have to be attended to with the ROV in shallow waters.
Mhm.
We haven't, knock on wood, so far
Knock on wood.
on this
Yeah.
dive, we've not had any of these real problems, so that's great. Yeah, I think we're in that thermocline. Colleen, do you have a? Sorry, you guys were discussing. Do you have an updated sense of a plan? What we're doing? Yeah, that looks like a little sponge. Good eye. Yeah, that white one. Does it have a top on it? Like a yeah? Yeah. Do you want to do a Solaris measurement there, Dr. Hansel? It's hard to see how big that is. Yeah, and the squeezer. We'll put the squeezer right in the top and fire it. One looks maybe the most approachable. Doesn't look mean. It doesn't look mean. All right, so we have a sponge in sight here. It's not super big, but yeah.
Yeah, come from off top for a few minutes. Thank you.
What'd you guess? That's a foot tall? Not even quite. Oh, you got the lasers on? Those are 10 centimeters, right?
Yes, those are 10 centimeters.
Yeah, so on the outside. Yeah, yeah, maybe even down further, you know. So that's going to be the water that it's pulling in. So we're going to compare that to what's coming out of the top. Yeah, that's good there. Yeah. Okay, that's the outside, Lena. We're measuring that now. Yeah, sort of stick it straight down the throat. Are we ready to do that? Are you ready to do that? Wait just one sec. The little squid right there. The red eye. Good? Okay. It doesn't need to go all the way in, but you know, so let's just say something like that. You know, submit. Yeah, you're ready. We're ready for you to try that now. Yeah. So it doesn't need to be fully fully stuck down. There you go. Is that in? All right, we're in the top. Okay, yeah, looks good. No, we didn't. It's not on right. Yeah. Yeah, it's definitely in there. That's good. Just hold that. It'll take a sec. What's the delay? 10 seconds? No, it's true. I know. Right. Oh, so the squeezer will tell us a little bit about that. Okay. Oop. Okay, we're out. Do you want to back in? I don't know. Yeah, what do you think, Lena? We're done? Okay, I think we can pull it out. So we're gonna want to do a squeezer and a Niskin here, right? A little bit. Yeah.
But we don't know if it's pumping. So, but if we take a squeezer,
Tell us something about the.
if we do a squeezer and a Niskin, and then we should collect the tape. This would be her, blood sample.
Okay. And for squeezer we can do. Oops, sorry. And so have you done the squeezer stuff yet? Okay. Yeah. All right. So 6 is done. 2 did not fire. Yeah. So we're gonna work with either 1, 3, or 1, 3, and this one is actually number 7 on there now. 1, 3, and 7. Feel free to just pick number 1 if you want. Or 3, or lucky number 7. Sure. Let's try 7. Yeah, so the approach would be grab the monkey fist and park it in the front on that ring, or that. Yeah, that's exactly. knows? Nope. Thought you were in there. What is that? Yeah. It kind of seems like it. I don't know. Yeah, so you know how this, yeah, open the jaw and then come into it and put it in the back of the jaw. Yeah, and then kind of close just to barely touching the white. And then we put it in position and squeeze the rest of the way. Sponge looks really small now compared to the squeezer. I can't tell. How did it get stuck? We haven't touched the number 2. Recent, yeah, not recently. Yeah, I think that should work. I think so. Yeah, as long as we keep it level. Yeah, we just need to make sure when we go to put it over the sponge. There you go. And now we want to basically, there's there's a nozzle on the bottom, a little intake. We want to, I know we can't see it very well. We kind of want to just position it right in on top of that. This is where if we had a little piece of hose on it. Something that could sneak in there. I know. Yeah, I was wondering if you come and then think that's what you're about to do. This is tricky. As much as possible. Hmm? Yeah. Okay. Whoa. I promise we're going to look for bigger sponges after this. Looks promising. There's a shadow. All right. Sure. Try to squeeze. There it went. All right. It fired. Piece of tubing on the end of that would have made that easier. Yeah. 7, but yeah. Oh, sorry. Yes, that was 7, right? Yes. And then, Colleen, I'm going to say that the Niskin 2 that we fired serves as this background for here. I don't think we need to fire another Niskin. Yeah. I don't know how you collect tissue on that sponge. It's not very big.
How do you think that compares to some of the coral size-wise? That sponge.
Once you grab one, I'll pass it.
Pop out.
Yeah. One coral right from here. Yeah, I've tried for the last like 2 hours to identify them, and I just can't.
Do I grab a piece?
Yeah, one left. Green puppy one? Yeah, okay.
Do you have any ideas of what it might be?
It looks kind of like it's in the Appenopathidae.
Mm-hmm.
It's one of the it's really interesting.
Yeah, it looks very cool.
It's also hard to see.
Mm-hmm.
If I have it, I can like measure it. Give it out.
Yeah.
All right, well done. See this green coral up here to the. We have a friend there.
Oh, hey, lionfish. That's a big one. Yeah, can I flag that as a video highlight?
Yeah. Sure. Flag away. Sorry.
That's beautiful.
So, does it have secondary stimulation? You focus on the one right in the center.
Oop.
So, could it be like tetra? Go ahead. Look.
Yeah. Mhm. Go ahead. Yeah.
Ben, could we zoom in on the lionfish, please? Can we frame it with the coral? Just because I think it would make a good video. Thank you.
It doesn't respond. Maybe it's on auto. There we go.
There we go.
Yeah, I
back.
think, Ben, do you want to?
That's
Can
so
you think
cool.
we just chop the top of the sponge off with the coral?
Mm. Yeah. Yeah. Yeah.
Here we go.
Insane. Oh yeah.
With the coral with the coral cutters, right? I think so. Yeah, I think it'll fit. So we're gonna put it.
He just chased that little fish, didn't he?
He's a bully. So we're just gonna give that sponge a haircut. Coral quiver. What is it? We're 11. Anything back there? 11's open. Sorry, what's that? Yeah.
Oh, he just got something.
Hello all, Jeremy Horowitz here to entertain you for the last little bit of this dive. It's been a very exciting dive.
Yeah, thank you for being here.
Oh, my pleasure. So we're collecting a sponge, and then we're going to collect a coral that's nearby the sponge. For the past 2 hours, I've been sitting here looking at the coral while they've been doing their experiments, trying to identify this coral, and I just can't. So it'll be really interesting to collect. It's one of like 4 genera, and I can't narrow it down any better than that without looking at it close up. So after the sponge, we'll collect the coral. And then I'm not sure what's next. We'll see.
When you're taking it back into the lab, what kinds of things are you looking at to help you identify what species of coral it is?
So the way that the branches are arranged, and then if there are secondary branches, how the secondary branches are arranged. So if it has just primary branches, it'll be one species. If some of the primary branches have secondary branching, it's another. If they all have secondary branching, it's another.
Okay.
And a lot of these genera only have one species representing the genus. So if it does represent one of these genera, it could be a new species, sister to whatever's already been described.
That's so cool.
So cool. And
Super cool.
Often on these expeditions, we find new species, so like it won't be so surprising to find a new species over the next few weeks.
That's exciting. I love how like nonchalant you are about finding new species. You're like, oh yeah, we just find new species all the time.
I mean, someone said it earlier that with ROVs, often what they normally like to do is go as deep as they can with the ROV.
Yeah.
You know, you buy this, however much an ROV costs. I don't even know.
A lot.
A lot. Let's say a lot. You're not going to go down, 100 meters. You're going to go down 1,000, 2,000, 4,000 meters. So we know less about biodiversity at this depth than we do at 4,000 meters because it's too deep to scuba dive
Yeah.
and it's so shallow for really advanced technology like ROVs.
Yeah.
But there's more species here than there are in the like abyss, for example. So we got a lot more to learn here.
Yeah.
And as you could see, like we don't have enough time to sample all of the things that we have right in front of us.
Yeah.
In this screen, we have 1, 2, 3, 4 different species of corals. And just out of this view, we have a whole bunch other, one of which we're going to collect.
Yeah.
And when we zoom out, there's one more black coral that has this like uniserial branching, so branches that are only on one side of lower
Okay.
order branches. I want that one too, but we can't stay here forever. So on a different dive, we'll grab that one.
Yeah.
It's just squeeze it. Maybe just squeeze it until it breaks.
It's a lot springier than I anticipated.
Yeah. Just take the whole thing. We've collected sponges before, and they tend to rip pretty easily.
This one certainly is not.
Certainly not.
I don't know if it's just me, but there's definitely, I'm looking at the sit cam right now. Can pull that up actually. There's a lot more colors maybe than when we first dove. Is that true? Or like, I know we're not really zooming in, but are you seeing anything new in terms of the encrusting stuff in the background?
Well, totally, because we were deeper on the last dive. So, the shallower you get, the more colors you're going to see, the more algae you're going to have. So this is all algae on the floor.
Yeah.
So very pretty.
Yeah.
And I think we are meant to go a little bit shallower. I don't know if we're going to do it today. Probably not, because we have nice. Yeah, I was just going to say, should we check? Yeah, we could spend days in this site and just barely move. What's the depth of it? Well, no, where do you typically find it? Mercio. So I guess on a different dive, you'll collect shallower and do like a bounce dive. Maayo Ben. are we doing on time? Is it okay? If you want to collect something more of your targets for now, it's fine. Yeah, I think the little fish are attracted to the lights of the ROV. So it's a little fish party.
They kind of look like squid, some of them.
Yeah.
I can't tell though. They're quite fast.
Oh, maybe you're right. Are they all baby squid?
Or shrimp.
Oh, there's some crawling you could see on the 12. All right. And the sponge is still there. It'll just grow back.
How fast do they grow?
Oh, I mean, 3. That was really helpful. But it will grow back, and it'll be fine.
Oh my gosh, there's more. It's a whole swarm.
Corals? Yeah.
Okay, so we're going to be looking for some full colonies, and with the purpose of keeping them alive. And we'll be videotaping that once we get them in the tanks. I'm sure Sophia's going to record it and share it somehow with everybody. I've never seen deep corals in the tank before.
That's very exciting.
I'm used to just throwing them straight into ethanol and putting them in a happy depth. So this will be really cool. And we've got a lot of corals to process when we come up.
We'll be working all night long.
We'll be busy.
Okay.
How many quivers have we filled up? Yeah, we'll do fine. I mean, it is the first time we're processing, and there's a whole lot of steps. We've got a lot of experiments with each sample, so it'll take some time.
Okay. What was the SH number on the sponge? So it's SH. Okay. Thanks.
So we aren't going up yet. We're going to spend a little bit more time looking for some full colonies. If we find, I think the target's Mauricia. If we can find one of them, we can collect.
Swiftia. Is that like another one that you're interested in?
Okay, so they've got targets in mind, octocorals. So yeah, let's try to find it together. If you see it, let us know.
Crowdsourcing.
That's right.
I guess off the bat, if I were to be looking for octocorals, like what would I be looking for, like as opposed to hexacorals?
Besides the vibe that you get with an
Okay.
octocoral, you
Just pure vibes.
Yeah.
but the number of tentacles. So octocorals, octo 8, so they have multiples of 8, so they could have 8 or 16 tentacles.
Oh, really? They can have 16.
Or 32.
Really?
Oh.
And hexacorals have 6 tentacles, or 12. Like a 16. Yeah. Divestream. Not to. I'm sorry. No, the. What's.
Whoa.
We found the coral.
It's very busy.
Alright. Nacella. We're collecting it. And, I mean, if you're just looking at this picture, there are so many corals.
Right.
Soft corals. There's a black coral right in front. There's actually a really thin black coral behind it. And then another black coral on the left.
I think that's a
Tennessee Nepenthes. Oh, actually. Ah, it's so interesting. Maybe Tennessee Nepenthes. I know. And so different. This. Yeah. So we're trying to grab the colony from the basal plate, trying to flick off the plate from the substrate to grab the whole colony. It's a delicate procedure, but it can happen. We could do it. So Ben is carefully extracting this coral from the substrate.
Mike is.
Oh, I didn't see who had the claws.
I guess it's teamwork. Oh. You want?
So we're just trying to decide which coral to collect. There's so many in the area. Got a bunch to choose from.
That one right now is just like, phew. Just in the nick of time.
Is he sweating in ROS? Alright, so Sam. Hard corals can have multiples of 6 tentacles, right? Like Tubastria. But so octocorals can have multiples of 8. Hexacorals, yes. Alright. More would be crowding? Yeah, I don't know. Okay. But what if the polyp is really big? The bigger, the more.
Yeah, I think that one is like the one we sampled earlier. Yeah, you can see the white balls and the reddish orange. That one is one of the ones.
Maybe a snake star wrapped around the coral that we're going to collect. This is Eliselidae.
Really?
Eliselidae. They're brittle colonies with elongate branches.
Go.
Look at a little squid.
The near-death experience. One coral.
So the crew gets fairly delirious towards the end of the of the dive. We're getting there.
That one, I mean, Mike was ready. Was about to trigger the jaw. Wait, actually, hold on. That one, no, thank you.
Yeah, from the angle.
Yeah, it's like the alien ship comes down.
It's like, yeah, nah.
Oh, actually, no, we didn't actually want you. We want your friend. Your friend, thanks.
Maybe they wanted to be probed in the lab. Oh my god.
Okay, here goes the collection. Alright, so in the description I have of this family, whip-like branches, like long, but this isn't. So is it that it just has a real wide diversity?
Which one? Do you guys want to? They're looking for feedback.
No, I got that. Okay, so when they're fans, they're not long branch-like, like elongate branches. No. Okay. All right. I think soft coral taxonomy is confusing. Right. So what you're not hearing from Sam is that it's not the macro scale features. Like we know what species are related to others, and although from like a macro scale, gross morphological standpoint, they might look really different. If you sequence the DNA and you find sister species, usually what's in common with them are these small features. In this case, the skeleton is the skeleton of this and other species that don't really look very similar. The skeletal structures are similar, and we're actually finding this with all across the Anthozoa. Branch species are related to unbranched species, which we thought wasn't the case. Very nice, beautiful.
Yeah. I thought that, yeah, it does.
So this is Elicididae. There's a few targets that we have in this expedition. Totally. I'm learning a lot. But in order to learn, you have to realize how much you don't know, which I'm now learning, how little I know. So our bio boxes have two corals now. A black coral we collected like, I don't know how many hours ago. And this soft coral. What is everyone's favorite collection today? I know what mine is.
The sponge squeezer. Jesus, look at that.
Our ROV is chock full of corals. So I think we're going to look for
Is this,
maybe
Colleen,
one
does
more.
that mean we're not taking any more samples or anything like that?
I think we're just collecting for the aquarium right now.
Okay. It would, all right. It would be great if before we hit the surface, we could even just test fire the rest of the squeezers.
Okay.
I know that would take a little bit of time, but just confirm that they work.
We already did 3.
Yes. Yeah, okay.
So you want to test by the other 3?
Well, I know it'll take time.
It's getting late.
All right. I don't know.
We're gonna keep trying to find another one of those types of corals, and also purple coral. So yeah.
Hey Luke, we're gonna start moving southeast. Alright.
Oh, what camera are people seeing? Is it the main? I don't know. So we're gonna cruise around for a bit. And the good thing is, we've been at this depth for the whole dive. So the corals that we collected are in water in a temperature that they're used to. So when we bring them up, they should be in good shape. Sometimes when you collect corals and you bring them up from the deep, especially after a slow ascent, they could look pretty unhappy. Their tissue could be sloughing off.
Are you guys seeing anything?
Yeah, I see that, Sam.
Yeah. Got a really good picture of tissue sloughing off a coral. I'll try to share it for the next time we're in the water. It looks like something out of Alien. It's pretty
lovely.
Have I showed it to you?
No.
Check it out. Pretty gross. So it looks like we're going to be collecting our last coral. We have recorded, we have gone live during the descent and ascent before. It might be possible to do. Maybe later in the expedition when things have been running smooth from start to finish. The blue water dives are pretty cool. You might recall when Falco was in Australia, we did blue water dives. So we spent like 5 hours just in the midwater, never seeing a coral. We call them the Dougal dives because Dougal Lindsay, jellyfish expert, would call in and spend 5 hours just identifying everything in the midwater. Those were pretty cool dives. Unless you wanted to see corals, then, you know, I was dying to see the seafloor. Okay, we've collected a big fragment. Looks like it's got some passengers on it. Some snake stars. Yeah, I mean, the ascent is pretty short from here. Maybe we could stay alive, I don't know, for a little bit. So right after this, we're gonna get all the samples out of these bio boxes, bring them into the lab. We're going to subsample all of them about 5 times for various experiments. Then, while those experiments are running, we'll image the colonies, and then we'll preserve them. And whatever's left over after subsampling will be sent to the Smithsonian Institution for long-term preservation. And for species identification, a lot of the times it's really hard to identify a species even when it's in your hand. Often you need to look at, for soft corals, sclerites, which are these small, actually it's what gives the coral its structure, these small features, these really small microscopic features. So you can use a scanning electron microscope to view them. And the same for hexacorals as well. Often you need to see the fine microfeatures. So we'll be confirming identifications after the expedition. The turnaround for the next dive, I'm not sure. I'm not sure. No, it's real cool because you get to see midwater. Well, like you see lots of like jellyfish, tunicates, metaphors. No, I see a lot of cool stuff. It's different. Yeah, I don't know how long until the next dive, but on this ship for these 3 weeks, we're going to have the ROV in the water as much as we can. So I'd expect a pretty short turnaround time. There's a question about, do we use the water that comes up with the corals in the quivers? And the answer is yes. That's the best water to use. So we'll bring the water in to the lab with us when we collect the corals. Got to ask an ROS person.
When the water comes up in the quiver, will you take an ROS sample of it to see if more ROS come from it being disturbed or no? This was a question on the stream too. It was in the comments. I thought it was a good question. Am I on here? Click it again, but
Talk. Colleen's going to get her microphone set up, and she will answer the question.
So is it? Okay. So looks like I'm on. All right. So the question about whether or not we're going to measure the superoxide inside the boxes or in the quivers is that's a great idea. We could do that, but normally what we want to do is try to get the corals out of the quivers as fast as possible. So we bring it up into the lab and we're gonna start processing them and get them into the aquaria, and then we will be doing measurements of reactive oxygen species in aquaria within kind of conditions that they'll be happy in. So nice cold water, dark conditions. So we'll be doing some of that in the lab. We'll also be trying to figure out, what is producing, why are the corals producing the superoxide. So we're going to be feeding them. We're going to do little wound experiments to see whether or not we get more production when they're wounded within these aquaria experiments, where we can really kind of target and control the conditions.
There's a question about seeing the lab. There'll be plenty of opportunities to see what we're doing in the lab. This will be the first chance we have at actually using the lab. We spent a lot of time setting it up for all these experiments. So you'll get to see them in use.
We don't. We could do it in the lab though. We could put it in one of the. Yeah, sure. Unless you just want to identify it for now.
No, we can put it in the lab.
Okay.
We have enough room.
Okay. Yeah, let's do a collection. We can put it in the bio box. Yeah, let's take it, I guess, from the base. I can't tell how big it is from this. Yep. Okay.
That's a cool one. Yeah, there's talk about the scientific claw game. Actually, our ROV pilots by law are not allowed to play the claw game because they're too good, and it just wouldn't be fair. Our pilots can do it even if it's rigged.
So this is 590, right, Sam? And we're calling this Swifty a question mark? Three A. Yes.
Oh, but this is not a red Swiftia, right? Okay. So we're not sure what this is. Little Selp wants to get in. Are you able to show the map to the people, like where we are? So in case you're curious, we have an inset map that shows where we've been today. And you can see we're at the top of this ridge where all the biodiversity occurs. So we've just been traveling along this ridge for the last 10 hours. So this is a map that I think we added a little bit to this map, but most of it is bathymetric data that has been made from previous expeditions. No, this is all us. Yeah, we did it all. Isn't it beautiful?
Mm-hmm.
So I think this lionfish has been following us for a while. Oh, now you like mid-water dives? Ooh. So we don't use AUVs on this expedition. All ROV. We do have a drone, but it's an aerial drone. So you might get some really cool aerial footage of us later on in the expedition. So the next shift is starting to wake up and join. Well, I'm also on the next shift. I just didn't sleep. I was delirious. I woke up and I was like, I think it's my shift soon. That was like 12 hours ago. So we'll see how we do on the shift. I'll do great. Totally. Yeah, there's so much excitement that it's really easy just to stay up. And like in this control room, there's so much going on. So many corals.
Sorry, Juliana.
Alright, well you guys have been with us for most of the dive, so we could all be delirious together.
I'm looking. I'm not seeing
So what is it?
the species we
Alien?
measured earlier.
So you're saying it's an alien? An alien. Yeah. So you can see a lot of diversity. Nothing new for this dive. We've been seeing this the whole day. It's quite beautiful. A lot of fish, a lot of corals, high diversity. A lot of snow, a lot of marine snow, which is food for a lot of these corals.
So it goes from 0 to 140 here, so it kind of maxed out at 100. This one goes about 400.
Well, I hope all of you guys are going to stay awake for the next dive.
Yeah, it's speed. Oh, it is. Yeah. And then the vector.
And that's north is the west.
No, we're still trying to find this purple. It's like a really deep purple. And it's kind of bushy. So it's pretty easy to spot.
Yeah. I don't really know how to interpret that though. Shallow versus. I think the sonar will send out in a thing it'll do shallower so it can kind of resolution so it'll be deeper. So this will probably be higher resolution and we're watching very deeper.
I mean, it makes sense. You know, what we see it.
Yeah.
It's something south.
Yeah, so.
All I see is yeah.
Yeah.
So like that's what's more enough.
Yeah.
Yeah, that's useful. Not there.
Yeah, I'll look here.
I kind of noticed.
Yeah.
Can we?
Yeah, you went there?
Yes.
Yeah. There's no
AUV? Right. We don't. There's no AUV. That's an automated underwater vehicle. so it's untethered.
I wish we had written down the depth
So
that we had
you
been
would
at
deploy
when
it,
we-
and it would
I
go
guess we
just
could go
above the
back,
seafloor.
Look at the details.
So it would map a short area, but with really high resolution. Here comes J-Rod. Ready to go. Do you need someone to do one of those things? There's enough people in the room, you know. Unless you want to drop one of them. So these coral whips colonies all look like soft corals. You could tell because they have that bend and then they kind of limp over, which is very typical, and this is a vibe that you get when you see them. So these are all octocoral whips. So we're still searching for another colony to collect. What's a cool flabellate colony in the middle of that balmy? Do you guys use the word balmy? Like a mound, a rock mound of coral? Balmy. It's an Australian thing, I think. Balmy. Right? Did you know what that word was before? I think I learned it in Australia. Balmy? No. But when we were in Australia, I think we were saying like go to that balmy. No?
Here.
All right. I mean, you don't have to believe me. I make things up. Are they talking about that flabellate colony? It's an interesting one. So we got some sponges and soft coral whips, and not sure what that flabellate colony is.
I mean, we'll be back down in a few hours.
This is a pretty octocoral dominated area. So you could see a lot of activity here as we travel shallower a little bit. We're at 140. Seeing lots of corals situated in ways that maximize their ability to catch nutrients.
We call it.
So these are all still octocorals.
Yeah.
Everything here, except for the sponges.
Okay.
Oh.
We got a lot of good stuff to get us started.
Okay, I think we're gonna call it a dive then. We're gonna come back down in a few hours after we've processed our samples and had a chance to put all our corals in aquaria and get them happy. And then we're gonna come back to a very similar site, a little farther east, and then look for some more corals to measure. Okay. Oh, here? It looks like a small one. Do we want to get one? All right. All right, dive's not anything. We're gonna just do a whole collection and put it in a bio box, please. No, no measurement on it, just a collection. I wonder what that dark part on that coral is. You see that?
Are you talking about this?
Yes.
Yeah. Do you know what this is? This the black thing on the coral? This is a very diverse spot. Covered. It's crazy. Yes, please.
He came out, he was like hiding in the ledge.
Yep. Yes, please. So, file box 2, A and B are open. Yep, both of those are open. Great, thank you.
All right, everyone. Colleen Hansel has left the room, and I'm just coming on here to sign off for the night. Thank you all so much for watching. As always, don't forget to subscribe. Hit the notification bell if you want to be notified for these live dives. We're going to have another one happen shortly this morning after all the samples have been collected and processed. And on behalf of myself and the rest of the team, we're really happy to have you here. And yeah, tune in tomorrow morning. We'll be back for sure for another dive stream. And I'll probably keep the feed on for just a little bit longer, but we don't have much ascent left, so it'll be on for just a little bit longer. But yeah, signing off now. Thank you all.